right. Well, hello, everyone. Welcome to our annual Muscle Study Group meeting. This is a very exciting day for all of us. We've all been looking forward to it. We're all a bit nervous about it because it's a new thing to do this all virtually, but it's just extremely exciting to actually launching this today. So Michael, Hannah, and I'm Rick Barron. I'm the chair of the Muscle Study Group. Michael Hannah is the co-chair. And we've got a few remarks that we want to say here to kick off the meeting. So I'll start. So on behalf of the Muscle Study Group, we would like to welcome each of you to the 2020 Muscle Study Group annual meeting. It's an exciting time in neuromuscular research as we continue to grow, and obviously we're adapting to COVID and other issues. This year is different because our meeting's virtual. And I believe it's an effective way for all of us to communicate at a distance. We've set this meeting up for 3 morning sessions. As all-day sessions, we thought would have really contributed to some, what I'm calling ZFS, Zoom Fatigue Syndrome. It's a new Barron acronym. And also our European colleagues, we want to let them sleep in the evening. And if we went all day long in US time, that would be a problem. So we've had such a huge response to this. You know, normally we get about 150 people. We have essentially 500 people signed up for this MSG event, and it's so popular, we're actually considering maybe doing another mid-year event. Dr. Hannah, I'll turn the next few slides over to you.
So I'm Mike Hannah, I'm in London, England, and I'm delighted also to welcome everybody to this MSG annual meeting, which is virtual. I want to thank this year's planning committee for putting together what we think is a really interesting and excellent agenda, covering a broad range of topics across the neuromuscular field. And they've spent a lot of time planning this and of course changing the course when we realized it needed to be virtual. As Rick says, it's fantastic that we've got as many as 500 delegates from across 20 different countries. So we'd really like to welcome all of you. So for those of you that don't know, the goals of the MSG are for it to be the premier organization and annual meeting for clinicians and colleagues interested in neuromuscular disease, and in particular developing treatments and trials. We have an important focus on supporting and developing young investigators in the field, and that's been really important to us and continues to be. We're keen to foster international collaboration for some of the rarer diseases. For trials, it's important we collaborate internationally. And also importantly, we want to connect effectively to preclinical investigators who are looking at potential pathways for new treatments. And also we highly value our industry partners, and a key aim of the MSG is to engage effectively with industry to support the development of drugs for patients. So we particularly want to thank all our sponsors this year, and we're really grateful for the major significant interest in this meeting. So gold level sponsors are Genentech, PTC Therapeutics, and Sarepta Therapeutics. Silver level is UCB, which is also Ray Pharma, now part of UCB. So thank you to them for silver level sponsorship. And bronze level sponsorship, Argenx, Biogen, Cytokinetics, MT Pharma, and Orphazyme were again very grateful for their bronze level sponsorship. Corporate level sponsorship, we want to thank Catalyst Pharmaceuticals and Spark Therapeutics. So it's really important that we have the support of those colleagues in industry, and we're really grateful to them. So here's a list of the executive committee of the MSG, and again, we're really grateful for all the time and effort they've put in. I won't read all of the names out, but you can see the names there from across the US and Europe. And these colleagues have done a lot of work to keep the MSG going and enable its strategic direction. I'd like to thank the outgoing Executive Committee member, Marsden, who's done a huge amount of work supporting the MSG and is now the MSG Treasurer. I also want to welcome the incoming member, Michael here from Vermont Medical Center. So welcome to him.
And so, yeah, I think I was going to do this slide then, Michael.
Okay.
If you can't tell, we figured out who was going to do what slides 2 minutes before we started. So, which is the typical, right? I mean, it's
You stopped me.
Yeah, it's nothing new. So we had a great MSG annual meeting planning committee. And this is the list of it. Carolina Barnett Tapia, Shafiq Karam, Gita Ramdahari, James Lilliker, Kim Hart, Lindsay Alfano, and then sometimes Hannah and I would weigh in, but they really did an amazing amount of work to get this set up, and we were able to pivot to go from an in-person meeting, it was supposed to be in Georgetown, to this virtual meeting. So we really owe a huge thanks from Liz Polk. And Liz, the group needs to hear your voice here at some point. Can you say hi? Are you muted?
Hello, everyone.
That's Liz. And she is, as you know, our administrative manager for the MSG. She does an amazing job. She works out of the KU offices where Mazen Damachki is, my old office. But today, Liz drove into Columbia, Missouri. She's in a room next to mine, so that we can be geographically sort of close to each other for this big event. And I'm in my University of Missouri new office, which I've been at since May. So the other big thanks is MRD Gil. Gil, can you say hello to the group? Is it easy for you to do that?
Hey, what's up?
That's Gil. He's a producer. Gil is in New York City. He's a real producer in New York City, and he's a Zoom meeting facilitator. And we are so happy to have Gil and his magic to pull this thing off. And then another huge help was Ayla McKaylee, who's one of the premier senior research coordinators, project managers at University of Kansas Medical Center, and she helped Liz a lot. So thank you, Ayla. All right, so this is what I am really excited about. So the volume 1, issue 4 of the RRNMF Neuromuscular Journal. So I printed mine out, obviously it's all electronic. And so the abstracts of this proceeding are in this issue that came out 3 days ago. So all of you can look at it, download it at the website here, journals.ku.edu/RNMF. And I think it really looks good. And I am hoping this will become the home for the MSG, and I'm working on the paperwork to get this all in PubMed, and that should be easy to do now that we've got 4 issues under our feet. It's already in, all these are in Google Scholar, and they have DOI numbers, so they're legit, but we'll get them in PubMed very soon. So thank you, and I pick out the artwork, by the way. It's been Rembrandt for the last 4 issues. Okay, MSG Fellowship Collaboration. As you know, this is a huge, exciting thing that we started doing a few years ago with Industry Partnership and with American Brain Foundation Partnership. So the industry gives MSG funds, and then AAN matches. We put in 2/3, they put in 1/3. And it provides for a 2-year clinical translational research fellowship, primarily aimed at clinicians. Well, the last 2 have been in the USA. It could be international as well. And so we'll get some fellows outside the USA. Next slide. So the first fellow was Reza Sayed-Dasaji. Oh, I'm sorry. This is, okay, this is our current fellow. It's not the first fellow. I'm sorry. this is right. Yeah. So Reza Sayedizaji at Mass General Hospital, proximal nerve visualization in CMT, and then the newest fellow that started in 2020, that's starting now, is Paloma Gonzalez Perez, MGH, molecular biomarkers of myotonic dystrophy type 2. I'm sorry, I messed that up, Reza and Paloma. But they competed for these fellowship slots, and we're extremely happy that there are MSG fellows. We had one fellow that's already completed at Baylor. And so this is very exciting. Next slide. Is that talk about the timeline? Yes. Yeah, so the deadline to apply is October 1st. So obviously, if you haven't applied, it's probably too late. You know, it's a real research application with a research plan and mentors and training plan. So while we are theoretically now accepting applications, I assume if you're going to apply, you've already done so. And then Michael and I attend the ABF selection session in a couple of months, and we'll pick our fellow that will start next summer. Next slide. And you'll hear from our current fellows during the proceedings over the next few days. Okay, Michael, after I flub that one, I'm going to turn this over back to you.
So one of the central parts of the MSG is to have MSG approved studies, usually clinical trial studies. And the advantage of this is the kind of stamp of approval and prestige of having essentially a peer-reviewed assessment of your study by the MSG. You get access to statistics support through Rochester, particularly Michael McDermott, who is a really superb and very experienced statistician in particularly neuromuscular diseases. You get access to the data management through Rochester and the support of the expertise of Rabbi Tawil. And we believe having an MSG recognized study will help your career. And of course, you get to hang out with us at the virtual meetings. Next slide. So this is how to submit a study for consideration of being an MSG study. You essentially email a copy to Liz of your protocol, a letter of request, including members of your steering committee. The steering committee will need to be appointed. You don't have to use the MSG Coordination Center, that's optional, but this would allow your study to be labeled as MSG. And what happens is the submissions are reviewed by the executive committee for approval. Next slide. So this was the last face-to-face meeting that MSG met in Salt Lake City, Utah last year. This is the photograph in the mountains. There's already, I think, twice that number on this call already, so that's impressive.
So we were struggling, Michael, on how to do a meeting picture. And at some point in the next few days, we're going to want everyone to turn on their camera and to take a picture of their screen, a screenshot, and then send those in to us. And I don't know, we'll figure out a way to collage it together.
Okay.
Liz and Gil can give us more on that as the meeting goes on, how we're going to do that.
Okay, next slide. So this is the MSG membership. So we now do this by individual sites as opposed to individual people. Application from a site is looked at by the executive committee. The site needs to pay $500 per year, and that would cover all the research personnel at that site. If people don't pay their membership for 5 consecutive years, membership may be dropped. Any site can join by submitting an application, and that's on the MSG website. And it's collecting by site rather than individuals. And you can get further details from Liz about that. But we're keen to have more members.
But you don't have to be a member, obviously, to enjoy this meeting. Right.
So these are the new members. We want to welcome these 4 new members for 2020. It's great to have them on board, and there are their logos. Henry Ford Hospital, Detroit, Virginia, Commonwealth University, University of Missouri, and University of Sao Paulo. So they're all very welcome.
Liz, did we vote in another one yesterday?
We just received for Vanderbilt.
Right.
Yes.
And so I know, yeah, so Vanderbilt will be the next one too. This just happened yesterday. So if anyone's on from Vanderbilt, welcome. All right, I just want you to know how we're able to pull all of this off economically. Obviously, we get money from donors and from dues, from pharma and from our dues-paying members. Where does that money go? It goes into a 501c3. So we had to create a 501c3 called the Muscle Study Group, Inc. And this is all the information about it. We recently made Mazen the treasurer since I moved 120 miles east to Columbia, Missouri. And we wanted him to be the treasurer there with Liz. And this is just basically the information. So that's how you do it in the United States. You have to do a 501c3, and this is where the money goes, so we don't have to pay taxes. That's the idea. All right, Michael, why don't you tell them what may or may not happen in 2021?
Yeah, so we were planning for the 2021 meeting to be in Georgetown and in Washington, D.C., which is a really nice venue. Has the kind of setup that we like to have, the kind of community spirit of the MSG. However, it's uncertain at this stage, and it may well be that this will have to be a virtual meeting, and same as this meeting. So we will let you know what the final plan will be. Okay.
All right, good. Now a couple of housekeeping rules. Everyone wants to know where the restrooms are, where the refreshments are located, and where the coffee's located. Next slide. You know where they all are. I don't know where your bathrooms are or where your refreshments are, but you all know. So this is virtual meeting. All right, I just had to put that in. Next slide. So if you would like to ask a question, click on the raise hand icon and the meeting facilitator will allow you to speak. After the sessions this morning, we're going to have a mingling session. Today and Saturday and Sunday, you will be able to go from room to room. Please stop by and listen, even if you don't have a specific question. Link to join will be in the chat, and this will happen later today. I'm really interested in how that's going to work. Gil's going to pull off some magic to make this all happen. This is a CME event, so it will be offered for this meeting, and you'll get the CME information soon. So again, it's just a thrill to have you here. I know we're supposed to have our first talk at 8:20, which is 2 minutes away. For the last couple weeks, I've been reading this unbelievable book. Actually, it's a quiz. Michael, what happened on November 28th, 1660 that would be relevant to the MSG meeting?
1616.
1660, 1660.
I'm sorry, I really don't know.
The Royal Society of Medicine was started. That was when Christopher Wren and Robert Boyle started the Royal Society of Medicine, not far from your office right there in Queen Square. Yeah, in a place called Gresham College at Bishopsgate. And
right.
So that was the first scientific society. And then they had the first scientific publication. And, you know, basically everything, all the scientific meetings since then have been iterative of that process. And so we're standing on their shoulders, as Isaac Newton said, but we're doing it in a new way. So it's just really exciting. Okay. Sorry, Dr. Hanna, I just thought of doing that this morning.
No, it's fine. It's fine.
So now we are gonna, initially we weren't gonna do any updates, but then we actually were able to create some time. And so we have 3 current MSG studies we're gonna get updates on, one that are scattered through the program. and they're all on IBM. And the first one is going to be given by my academic partner, even though we're now 120 miles distance from each other, but we're still academic partners, Mazen Demachki, professor of neurology at the University of Kansas and the executive vice chair of the Department of Neurology and many other leadership roles there. So Mazen is the leader of this really exciting phase 2-3 study of arimoclomol and IBM. And he's going to give all of us an update on where we're at in that international project. Dr. D.
Thank you, Dr. Barron, for this kind introduction. And I really want to thank the organizing committee for allowing me a few minutes to talk about this exciting study, the phase 2-3 study of arimoclomol in inclusion body myositis. It's an honor and privilege to present it to the MSG audience today. This has been an exciting journey. It started many, many years ago, but I'm just going to give you a snapshot of where we're at today and why we're here today, which is in a very good place. So I'm here really preaching to the experts in the field, and excuse me for just reminding us about some of the pathophysiology of inclusion body myositis, being both inflammatory from a pathologic standpoint as well as degenerative, with evidence of a mitochondrial lesion too. And some of the inflammatory approaches, however, have not been so successful. So the focus that we have is on the degenerative component as evidenced by vacuolar changes, amyloid deposits, inclusion bodies, as well as deposition of several different proteins, particularly B62, TDP43, and so on and so forth. So there's a natural ability for the body to fight protein aggregates and process them. And this inability of the body to do so results into aggregation of protein within cells. And we believe this is part and parcel of the dysfunction that's seen in typical muscles of IBM, where it manifests clinically. In addition to its diffuse distribution, it does particularly affect the forearm muscles, the deep finger flexors, dysphagia at the level of the bulbar musculature, and quadriceps muscle preferentially. There are other muscles involved, as we all know, the gastrocnemius muscles, the soleus, and so on and so forth. But certainly these are some of the main targets of the protein aggregates. The heat shock protein response is a normal defense mechanism that is kind of amped up and increased in times of stress. And that heat shock response is, this natural defense can be triggered across the whole body. It's not specific to muscle, in other words. And this results into the increase of different heat shock proteins, including heat shock protein 70, heat shock protein 90, and other chaperones. And the idea behind that is taking those folded, misfolded aggregates and correcting the aggregation, or targeting proteins for degradation and promoting lysosomal health altogether. Arimoclomol is such a molecule that works different from other medications, where it amplifies this natural heat shock response that's activated in physiologically stressed cells. And that's an important point, because arimoclomol does not have an effect on unstressed cells or non-stressed cells. So therefore, we feel and believe that it specifically targets, arimoclomol specifically targets the degenerative components of inclusion body myositis. This is this mnemonic of team together, everyone achieves more. And this has been a long journey getting to this point to present to you today where we're at with the study. Just want to remind ourselves of the journey to today. Along the way, many people ask me, why do you keep doing this? Why not give up? And I think that's important, not to give up or give in when it comes to important programs like this one, or any research project at all, I would think. Tenacity is really part of it. But then it's important to really get this industry academia collaboration, meaning from an academic standpoint, we have a target. We want to move fast. We want to do everything yesterday. But at the end of the day, the collaboration between academia and industry will lead to a better outcome. And what kind of outcome is it we're looking for? Though we had an FDA Orphan Product Division funded grant, we wanted to make sure not only access to the drug and placebo was available, but there are some operational bells and whistles that are essential as part of GCP compliance for any regulatory agency to look at data after the fact to determine if the study is positive, whether the rigors of GCP were followed. And so that was an important task that we kind of engaged with Orphazyme in collaborating with them to make sure that we have that very well in place so that we can, as we interact with regulators, feel confident that our interactions would be fruitful down the road. There were some add-on studies with pharmacokinetics, impression of change from the patient perspective, very importantly, but also the clinician perspective, MRI of the muscle study, sub-study, and an open label extension study now that's available. And I'll update you on where we're at with that too. The goal ultimately is, if the study is positive, to be well positioned for marketing approval. So along the way, we had to be flexible. We were funded several years ago, but we had to delay the first patient in until we got all those mechanisms in place, which was fundamentally important to the current success we have so far in the study and hopefully future success, and then invest extra time and energy into that in order to move the field of IBM forward. So this is the study of arimoclomol in IBM. It's the target is to enroll and randomize 150 patients, randomized 1-to-1 to arimoclomol 400 milligrams POTID versus placebo TID. There were some requirements as far as inclusion criteria. This is a very summarized inclusion criteria to make sure the patients fit the ENMC 2011 criteria for IBM and were not too weak to enter into the study. As part of that study, we're also doing a validation study for the IBM FRS. We're having also exploratory biomarkers, not only from the MRI standpoint, but also blood biomarkers. So the status update of the study is that the trial is fully enrolled as of May of 2019. The first patient was dosed in August of 2017. The last planned visit will be January 2021, with 152 subjects randomized at 12 sites, 11 in the US, 1 in the UK. And these are the primary endpoints. The IBM FRS is the primary endpoint. The secondary endpoint is the 6-minute walk test with 2-minute distance captured, the modified time up and go, manual muscle testing, muscle strength, isometric contraction of the bilateral quads, and grip strength. So of course, we all navigated research through COVID-19, as most of you did. The study remained strong and ongoing. There was strong communication between the Orphazyme team, the Premier team, and the academic team in making sure we address all the obstacles that would be encountered with COVID-19 and resolve them, where we did so successfully. And we preserved the integrity of the clinical trials. The main focus is to maintain subject safety and data integrity at the same time. So during the pandemic, a lot of the in-clinic visits were converted to remote visits that could be performed via phone or video conferencing. Home nursing services were employed and recruited so that local labs can be done by a home health nurse or technician. And the shipment of the investigational medical product directly to the home of the patient was enabled. And it's great that we don't have a metric, a primary outcome that's dependent on an in-person visit. The IBM FRS can be easily captured through a over-the-phone visit. So the trial is continuing despite the challenges posed by COVID-19, and this collaboration between academia and industry is strong and working extremely well. And now we have more than half the subjects rolling into the 20-month open label extension. And we hope to show results by next year, certainly by mid-2021, we should have results of the randomized control trial available. So with that, I'll stop here. I know I'm at the 10-minute mark. There may not be time for questions, but thank you for your attention.
Okay, let's turn things over to one of our moderators for the morning, Carola, to introduce our first speaker.
Oh, sorry. Bill, do we have the slide for?
Yeah, while we're getting those set up, I realized that we put all 3 IBM updates back to back this morning, so that which really is going to make this a great session. So thank you, Dr. Damatsky, for leading that. And go ahead. Who's introducing Dr. Khan?
Was supposed to be Carolina, but Dr. Barron, you can.
All right, I'll be glad to. So now we're going to have Dr. Khan from Queen Square in Dr. Hanna's group. Ala, you're going to give us the update on the Genetics Consortium. Very exciting. Thank you for doing this morning, or this afternoon.
Thank you so much. So, hi everyone. I'd just like to start by saying it's a great pleasure to present in this meeting. My name is Ala Khan and I'm working on the genetic of neuromuscular.
Sorry, excuse me. Before you begin, your presentation isn't up yet. We okay.
Is it up here now?
Not yet.
Yeah, press Alt Tab to switch windows, maybe.
Alt Tab. Is it?
Finish, yeah.
Try stopping share screen and then reshare the screen.
All right.
All right. Share screen.
Okay, and then go into presenter mode. All right, there you go.
There you go.
Perfect. Sorry for this. So my name is Ala Khan and I'm working on the genetic of neuromuscular UCL Institute of Neurology. And I'm here today to give an update on the MSG International IBM Genetic Consortium about what we have already done and the number of samples we have collected so far and our future plan with this study. So IBM is multifactorial in which the primary pathogenesis is unknown. However, it's believing that genetic factors are contributing in disease process. And by using several genetic approach, this will be help us to investigate several genetics pathways involved in IBM disease. Therefore, an international collaboration is ongoing to create a large IBM biobank to conduct this study. We are greatly thankful for every collaborator send us samples and who are gonna to send us more samples in the next few weeks. This study will help us to reveal important pathway in IBM and also to identify risk factors that may increase our understanding of these disorders and identify potential therapeutic target. As you know that we've already looked into APOE TOM40 genotype and found disease modifying effect. And also we've investigated the rare variants in VCP and P62 and found a rare variants that suggest mechanistic overlap with neurodegenerative disease. And also in the past, we've identified this significant MHC and SLZ variant using our exome data. But as you know that exome data is not very good for common genome-wide variants, so that's why we are now moving to GWAS. And we are expecting at least to pick up the MHC as a positive control. And MHC, as you know, remained the strongest genetic risk associated with IBM. And recent study by using immunochip array identified also amino acid position in HLA-DRB1 in IBM cohort, and also showed evidence that other autoimmune gene associated with IBM. We have collected so far 828 IBM samples, and we are expecting more samples to come in the next few weeks from Dr. Chris from USA and from Germany group and Sweden as well. And by this, we will have the biggest IBM cohort in the world. And this study will be ongoing, and we are also encouraging to collect more samples of IBM and increasing this number. Also, UCL Institute of Neurology will continue to fund blood samples and shipment. And by using this biggest cohort, we are aiming to conduct the first GWAS study on the biggest IBM cohort. Also, we found GSA array is the most appropriate approach to study this in terms of cost, and also to study the HLA region. And also, we have already got ethnically matched control run on the same GSA platform. We would like to thank every site sent us samples, including cohort, that we will receive in the next few weeks. There is still time to send more samples if any one of the meeting participants would like to send and to be part in GWAS study. And finally, we would like also to thank MSG International IBM Genetic Consortium and National Institute of Health for giving us control cases to conduct our exome association. Also, thanks for all patients and their families for participations in this study. Samples collection is ongoing, and everyone is welcome to participate in this study. Thank you so much.
Stay at the outside of the line. Thanks.
Carolina, are you there now?
Yeah,
Okay.
I couldn't unmute myself before, but now I can talk. Are there any questions? I don't see anything coming from the audience. So we have time.
Oh, no, the question was not about her talk, was what about Italy? Never mind.
Yeah, that's a common thread. And no raised hands. So if that is the case, we may as well have a few minutes early, and we can put up the next slide up. And while we have it up, I'll present our next speaker, Dr. Tassim Motafar from University of California, Irvine. And he will update us on the study named Influence of anti-5C1A antibodies on disease progression, clinical phenotype, and blood and muscle biomarkers in sporadic inclusion body myositis, a prospective evaluation. Tassim? I saw him earlier.
Said I cannot unmute myself. I wonder what the problem is, Gil.
He's on.
Okay, well, in the chat room.
Yeah.
Tassim, can we hear you?
Yes. Can you hear me now?
Yeah, good.
Yeah, I think it's a control thing. So it takes a little bit of a latency to give the control over for video or for audio. But good morning. Nice to see everyone again. I wish this was in person. I'm getting a little sick of being home, but great. So I was going to present an update on what's happening with the proposed natural history study that we've been working on for the last 3 years. And I'm sure you guys are sick and tired of listening to this, but we titled this study as Influence of anti-5C1A antibodies on disease progression, clinical phenotype, and blood and muscle biomarkers in IBM. And this was meant to be a 2-year prospective evaluation. This was based on some of the data that we had published back in 2016, where we showed that individuals with IBM who were seropositive had significantly more severe proximal muscle weakness, had more respiratory evidence for more diaphragmatic insufficiency, required more assistive devices compared to seronegative, and had significantly more dysphagia compared to seronegative. This was sort of confirmed in aspects of this was confirmed in a larger study run by James Lilleker with the European Consortium, where they not only confirmed some of the finding, but had a very interesting finding of reduced life expectancy in seropositive patient by a mean period of about 8 years compared to seronegative. We've recently looked at this in our own cohort of about 80 individuals with IBM, and have shown that there is a similar reduced life expectancy in seropositive patient compared to seronegative patient. And this was work in collaboration with Chris Weil, although this seems to be cohort specific rather than a overall effect. There are other data that have emerged by this very interesting study from Tawara's group in Japan, where they took serum from patients with inclusion body myositis and then passaged it into either mice or in myoblast cultures, and showed that only serum from antibody positive IBM had the pathological effect of creating P62 positive vacuoles in the muscles of mice or myoblast cultures in vitro on that. So suggesting that there may be a effect of these antibodies on disease pathogenesis, and suggesting somewhat provocatively that maybe the inflammation may be driving the degeneration rather than the other way around. Why do we need a prospective natural history study in IBM? And the reason for that is that now we are getting into an era of therapeutics in IBM. We still don't understand the disease fully. We don't understand the rate of disease progression, whether it's uniform or not uniform. And I would draw parallels to ALS, where we know that, for instance, ALS has fast progressors and slow progressors, and there are a number of factors that influence ALS disease progression on that. We don't, there are a number of biomarkers now associated with IBM, including the antibodies. There is a very large percentage of variant T cell population present in up to 75 to 80% of patients. And we don't know whether that influences disease behavior in any way or not, etc. And then there are certain aspects of the disease, especially the bulbar functions, that we really don't have a good handle on in terms of disease progression as well. Unfortunately, the kind of observational studies that we are proposing cannot be done in a context of a routine clinical trial, and needs to be done as a standalone observation study. So we proposed a consortium of myositis centers in the US, and then we proposed a prospective single-blind non-interventional observation studies with 4 specific aims. We have a steering committee led by Dr. Barron, and with Dr. Dimashki, Dr. Weil, and Laura Herbelin on it. We proposed 4 specific aims. The first aim was to determine whether anti-5-7A antibodies mediate disease progression over a 2-year interval in patients with IBM. The second aim was to perform a detailed morphological, histochemical, and histochemical analysis of fresh muscle biopsy specimens obtained from a subset of these patients. Aim 3 was to characterize the distribution of immunosenescent lymphocyte in circulating blood from patients with IBM, and aim 4 was to quantify the decline in respiratory functions in IBM. So there are strong rationales for each of these hypotheses. We hypothesize that the disease progression is going to be worse in IBM patients who are seropositive compared to seronegative. And based on some of the preliminary data, the reduced life expectancy, etc. But the other question we wanted to study as part of this was, what are the optimal outcome measures in IBM? And there still are questions about 6-minute walk test, IBM FRS. My personal favorite is time get up and go, which is usually not used much as an primary outcome measure in IBM. So we wanted to test those as part of this aim. So what we proposed was 6 monthly assessments starting from baseline. So a total of 5 time points over a 2-year period, with each time point doing a detailed motor and functional assessment, dysphagia assessment with a quality of life assessment, antibody and flow cytometry, as well as physical and neurological exam. The second was a muscle biopsy, and we realized that using archive muscle was probably not the right thing to do because of the length at which these muscle biopsies were taken from the time of the study. So we proposed a sub-study of about 50 patients who would undergo fresh muscle biopsy along with a concurrent blood sample, so we can actually compare the blood flow results with the muscle immunostaining on that. And we were mainly interested in these lymphocyte, ovarian lymphocyte population, in addition to the pathological features. There's been some suggestion that there are pathological differences even between seropositive and seronegative IBM cases, with seronegative cases having more vacuoles. And there are differences in COX negative fiber distribution as well. Particularly interested in CD57 as well as KLRG1 lymphocytes, which are usually present in high numbers in patients with IBM. But additionally, we were also interested in these particular subpopulation of cells which express Kv1.3 potassium channels. These are shaker type potassium channels, and there are drugs that are available. So we wanted to use that as a muscle biomarker as well. The hypothesis again being that these T cells at some point become unresponsive to conventional immunosuppressive therapy. So we need to understand a little bit more about the features and the distribution of these T cells. So these are the stains that we were proposing to use. They're all going to be centrally read. And rather than sending muscle tissue, what we were hoping was that each site will do the biopsy, freeze the biopsy, and then cut sections and send the sections to us for staining purposes. Hypothesis 3 was to look at the immunosenescent lymphocytes, and as I said, they are very interesting immunophenotypic changes in circulation as well as in skeletal muscle cells. These have the appearance of chronically activated immunosenescent cells with loss of CD5 and CD28, with gain of CD57 and KLRG1, and they have features of effector memory T cells, especially the TEMRA cells, which are the very advanced, highly differentiated cells. So we've worked the gating strategy. We've really sort of fine-tuned the flow strategies for this, and we are able to reliably show all of these features in large sample batches. We can actually do up to 12 antibodies at the same time, gating on the T cells. And we've shown that about the incidence of about a KLRG1 is up to about 80% in these samples, and CD57 is about 65% on these cells. And we also interested in not only in all of the lymphocyte, but actually selecting a subset of lymphocytes that also express CD137. CD137 is a marker of recently activated T cells, with the idea that if we see a higher distribution in CD137 positive cells, that may suggest that these cells are responding to some recent infection or recent antigenic stimulation rather than a chronic stimulation per se. So the outcome measures were to assess the frequency of CD4 as well as CD8 positive KLRG1 cells, explore any relationship between these markers and seropositivity to NT5 antibodies, to look at KV1.3, and then we also will have serum from them. So we decided to look at the 10 most frequent cytokines as part of the analytes and look at their relationship to disease behavior. And then hypothesis 4 is respiratory failure. We've been interested in this. We routinely do pulmonary functions in an IBM patient and have been impressed with reduction in IBM related pulmonary function. So we wanted to characterize this a little bit further in this study with looking at FVC, MIP, and nasal inspiratory pressures, as well as FEV1. And we have some preliminary data suggesting that there is a progressive decline per se. The power calculation suggests that if we need about 150 total number of subjects, and with the distribution in the US of about 66% being seropositive, we think that we need about 100 seropositive and about 50 seronegative to be able to make good statistical measures and analysis. The statistical part is going to be done locally here at UC Irvine. We have Professor Bin Nan, who's a statistics professor who's had a lot of experience with cancer studies, etc., who'll be doing the statistical analysis. Data management is done by the muscle study group led by Dr. Taville and through Rochester. Kansas is going to be the central IRB for this study. We have a consortium, as I mentioned, of 13 sites. They are strategically placed all over the US. And all of these have adequate number of IBM patients and experience with IBM studies. So we think that recruitment should not be an issue. Each site is only expected to contribute about 12 subjects for this study. As part of this workup, as part of this grant planning, we did a survey of IBM patients through the Myositis Association, specifically asking whether they would be interested in participating in a non-interventional study that may require frequent visits and even muscle biopsies. And we got an overwhelmingly enthusiastic response. This paper was published recently in the Journal of Clinical Neuromuscular Diseases. And then we did something that Dr. Barron is very familiar with. This is a concept that came out of the National Center for Advancements in Translational Sciences, the concept of having community engagement studios where we get the stakeholders around a table and ask them what's relevant to them and what should we do to help design the study better. We also did a quality by design studio, which is a concept that came out of the CTTI, which again, I know Rick Barron is very involved with that, where you look at essential elements of the study design and critically analyze it as part of that as well. The grant was scored quite nicely. It got an impact score of 25 with a 9th percentile. Unfortunately, it's waiting for the budget approval for next year's NIH budget. And I can't get a straight answer from the program officer whether it'll get funded or not. My fear is that because of COVID, this may become a victim of the COVID infection, and NIH may decide to withhold funding for COVID-related causes and not fund this one. But we won't know till end of October. Wishful thinking and positive thinking, if it does get funded, we are hoping that we can have a study start somewhere in January or February of next year, with the first patient enrolled in April. As I mentioned, Kansas is the smart IRB, so we have a central IRB for this. We anticipate recruitment over a 12-month period, and the study obviously will be finished by 36 months at that point. And we give ourselves enough time at the end for data cleanup and analysis. This is provided that NIH decides to fund the full 5 years. Lately, they have been funding 4 rather than 5 years. So we'll see how this evolves. That's all I have to present. I'm happy to take any questions or any criticism or comments.
Okay, I think we're coming back to time. We have time maybe for one question or two. Are there, I don't see any raised hands. No questions in the Q&A. I would encourage all of attendees to don't be shy and do ask questions. I know the virtual setup might be a little bit overwhelming, different to what we used to, but one of the main things that I love about Macro Study Group is the ability to chat to each other and to get feedback. So please do send us, even by Q&A, if you don't want to talk over the mic. Nope.
So, Tyson, you said you got you verbally said you got to score 25, but your slide said 20.
No, that was an error. So it was an impact score of 24 with a ninth percentile.
Okay.
So and their funding pay line last year for NIAMS was 11. Now what the program officer is telling me is that because the budget is more than 500K a year, it goes to a special decision committee at the institute. So that's going to be end of October.
Well, you've been very tenacious with this, and I bet you'll get it funded.
Well, I mean, the bigger question would be if it doesn't get funded, should we even bother to resubmit or not? There's a question from Tom Lloyd about enrollment in natural history study. I mean, the expectation would be that they would stay with the natural history study for the length of it. And as I said, for each site, there will only be 12 subjects per site. So for a site like Hopkins, which clearly has more than 80 patients, the hope is that they will select patients who will stay for the entire period of the natural history study, because if they go into clinical trial, then that's going to influence the natural history study. All right, I guess there are no questions, Carolina.
Okay, thank you, Taksim. Just a couple of minutes before we start, a minimal housekeeping. Please leave the chat to send suggestions, comments, and say hi to each other. The Q&A for actually asking questions. There's a great suggestion by Geeta for all of you on social media to use the hashtag MSG2020 to see if we can get the meeting trending. So I would encourage everyone, hashtag MSG2020. And please do use the Q&A for asking questions. And we're right on time, so I'm going to move to our next speaker. And part of what we wanted this year was to get ideas from different perspectives and different diseases about things that we can implement in our neuromuscular research. And implementation science and how to bring new technologies is a main issue that we're facing with this new treatment. So I'm very pleased to present Dr. Abby Fisher, who will talk about development of an app-based intervention to promote PA in people living with and beyond cancer. Thank you.
Hi there, and thank you very much for inviting me to talk. I can't pretend I wouldn't rather be in Washington in the fall, but I'm in my living room in London, so that will have to make do. So I'm based at the UCL Department of Behavioural Science and Health, and I'm going to talk about some work we've done as of developing an app-based intervention for people living with and beyond cancer. So this is my group here. I lead the Energy Balance and Cancer Research Group, and our overarching aim is to use behavioural science to support people living with and beyond cancer. And we specifically focus on the energy balance behaviours, so physical activity and diet. This is some of my team here, and I want to acknowledge in particular Anna Roberts, who led some of this work as part of her PhD, and our funders as well. So some brief background to this work. There's a very large body of randomized control trial evidence suggesting that physical activity is safe and feasible during and after cancer treatment. The evidence is strongest in solid tumours, particularly breast cancer, but also prostate and colorectal. And there's really strong evidence for improvements in physical function like fitness and psychosocial outcomes like quality of life. And in terms of cancer-related fatigue, which is probably one of the most debilitating and most commonly reported side effects of cancer treatment, there's meta-analytic data comparing physical activity interventions to pharmaceutical or pharmacological treatments, and actually showing that exercise is more effective and should really be the frontline treatment for cancer-related fatigue. And we also know that exercise improves management of some of the common comorbidities we see in cancer patients, like cardiovascular disease and diabetes. So there's enough evidence for the Independent Cancer Task Force to recommend that physical activity interventions should become part of cancer care and be offered to all patients. So in order to do that, we need interventions that are evidence-based, accessible, and also feasible for implementation in our healthcare system, which is obviously challenging, but kind of underlines the work that we do. So the work I'll talk about today fits in the Medical Research Council framework for developing and evaluating complex interventions. And the first part of the work fits in this early development stage. So particularly doing formative work with our user group. So the overarching aim of the work I'll talk about is really to test the efficacy of an app-based intervention delivered as part of cancer care. But the work I'm going to talk about really is to describe the formative intervention development work in the hope that some of these methodological approaches could be applied to other clinical populations. So this is an outline of our intervention development steps here, and I'll go through them one by one. But firstly, we systematically reviewed and meta-analysed existing app-based interventions. We then wanted to determine the interest in our target group for this type of intervention. We also wanted our target users to try some of the existing publicly available apps. We wanted to understand how clinicians felt about recommending physical activity apps as part of cancer care. And this led us to develop our intervention materials, and then we are conducting a randomised control trial of these. So firstly, the first thing we did was systematically review and meta-analyse the data of existing interventions that were out there. And if you're interested, our paper's available to read. But the rationale was we really wanted to see what had been done before and whether there was any indication that this mode of delivery was actually effective. So we included any digital intervention that targeted physical activity. We also included sedentary behaviour and diet as well, but I'll focus on physical activity today. We included adults with any cancer diagnosis, and an early scoping suggested that there wasn't going to be very much out there. So we included randomised control trials and pre-post interventions as well. And to briefly outline the findings, we saw a significant increase in favour of the intervention compared to usual care. So digital interventions, there was some evidence of efficacy of about 42 minutes per week. And when we looked specifically at randomised control trials, this was about 50 minutes a week. But in terms, when we did this review, most of these interventions were delivered via a website, and there wasn't very much app-based interventions. Like, wasn't very many app-based interventions out there, and actually there were only two, and one was conducted by one of my master's students. There was high variation between studies, and they all had a high risk of bias. So they all used self-reported physical activity, which is obviously prone to recall bias. There was definitely a need for larger, high-quality randomised control trials, but we thought there was enough evidence of effectiveness to make this worth pursuing. So the next stage was we wanted to determine interest in apps in our target population. So there's a kind of perception that older adults may not be interested in app-based interventions, and we just wanted to know whether that was the case or not, and whether there was interest in our target population. So we worked with 11 hospital sites across London and Essex in the UK, and we sent out about 14,000 health and lifestyle after cancer surveys. And the survey contained questionnaires about diet, physical activity, other patient-reported outcomes. And also, we asked patients to report whether they would be interested in an app or not. To be included, patients had to have been diagnosed with breast, prostate, or colorectal cancer between 2012 and 2015. Our response rate was 43%, so we had about 6,000 surveys. Majority were breast. The median age was reflective of the cancer population, and slightly more males than females, majority white, and a mix of education levels. So the main finding was that around 25% of our target population expressed at least some interest in an app. And there were some demographic factors associated, but the association with age was very small. There was a small association with ethnicity. People that used the internet were more likely to be interested in an app. People that generally wanted physical activity support were significantly more likely to be interested. And people who were more active were slightly more likely to be interested as well. So with all the caveats in the world, this cross-sectional survey, self-reported behaviour, also quite a select sample. So we only had a 43% response rate, and particularly in terms of ethnic diversity, it definitely wasn't representative of the demographic in London. And there was some interest in our target population, so around a quarter were interested. And we do believe that this is likely to continue to grow with increasing smartphone ownership in older age groups. For example, there was a study conducted in the US in 2010 in a similar population showing that only 9% were interested. So it's growing year on year. But we also believe that a one-size-fits-all physical activity intervention will never be realistic. And we're interested in whether we could optimize an app for people who wanted to use one. So this led us on to a study trying to determine, get some of our target users to try existing physical activity apps. And the rationale here was there are hundreds of thousands of apps out there already. And if one was feasible to either use or adapt, that obviously has sustainability, cost, and probably quality benefits as well. So we conducted a qualitative user experience study, and if you're interested in this methodology, of course, you can look at our paper. But we recruited 31 patients who had completed curative treatment for breast, prostate, or colorectal cancer. And we randomly allocated them to try 2 of 4 publicly available apps for 2 weeks. So it wasn't a test of efficacy. We weren't interested in increases in behavior over that time, but we thought by allowing users to try the apps, we'd get much richer qualitative data. So we interviewed them over the phone and thematically analysed the data. And the apps we chose were here, and the reason we chose these 4 apps were they were available across a number of platforms, so Android and iOS, for example. They also covered a lot of different formats, so we had tracking, we had strength-based exercise and video demonstrations, we had gamification. And also we're really lucky to work with a number of excellent physiotherapists who checked that the apps were going to be safe for our target population. So sort of briefly, some of the feedback from the apps were that multiple factors affected engagement. Some of the advantages the users perceived in this type of intervention were convenience, confidence building, choice. They were concerned about lack of supervision and safety. They did have some technical issues, so our researcher would have to call them and talk them through downloading the app. The perceived relevance of the app was really important, so they had to feel it was tailored to their ability but also their cancer experience, and they felt that any app-based intervention would have to account for some of the comorbidities that they faced as cancer patients, and also identity. And this is just one quote, but we saw many like these. So this user tried a strength-based program and found that things like press-ups and planks they just felt like a joke to her, and they were so difficult that she found them demoralizing. They also gave really useful feedback on some of the features. They really liked video demonstrations. They really liked some of the behavior change techniques like goal setting, self-monitoring, and feedback that we know are kind of really important for sustained physical activity change. Some really liked reminders and push notifications, some hated them. They really didn't like gamification. And they were quite polarized in terms of social support. So some really felt they would benefit from it. Some really liked the fact that they could exercise alone and thought they really would never take part in group exercise. The type of behavior they were most interested in was walking. They felt it was safe, accessible, achievable. And they felt very strongly that in order to take an app up as part, that it'd have to be integrated as part of cancer care. So they felt generally positive about the idea, and they felt that it should be offered before, during, and after treatment so that they could take it up when they felt ready. But they really felt that they'd need some kind of health professional badging, and a recommendation from a health professional would be crucial for them. And in this case, the cancer specialist nurse they felt was the right person to recommend this. So this participant expressed and sort of mentioned that they build up such a close relationship with the specialist nurse who's in charge of their care that if that person recommended physical activity, they'd be really inclined to do it. So we thought that was interesting and promising, and we were then really interested in trying to gather information on how specialist nurses might feel about delivering this type of intervention and care. And in general, they were very positive. We interviewed 19 nurses and got a lot of really rich data, again, which is available in our publication. They felt that there was a lot of benefits to apps. So in terms of their own skills and knowledge, they felt being given an app with some additional resources would allow them to get over some of the barriers they face in clinic, like time, lack of confidence, lack of referral pathways. They were concerned about their patients' sort of lack of digital skills and whether they'd be able to handle that type of intervention on their own. They really felt that the app would have to consider some of the comorbidities commonly seen in cancer patients. They really liked the idea that an app could help promote self-management and increase choice. So often they talked about the fact that some of their patients just would completely disengage with the idea of a group-based intervention, but would really be interested in being able to exercise alone. They also talked about issues and being kind of worried about the lack of supervision. And they felt that if they were to integrate it into care, they would need specific guidance and resources, and they'd also need an evidence base. But in general, they were positive. So while we were doing this work, Public Health England, which is an agency of the Department of Health in the UK, released an app called Active 10. And we thought this ticked a lot of the boxes that both the patients and clinicians had identified. So it targeted walking, it included goal setting, self-monitoring and feedback. It had the branding of a public health agency. So we went back to our original qualitative users and we asked them to try this one, and they were generally positive. And we then developed some additional materials, so resources that could be given with the app in clinic as the clinicians had asked for, as well as trying to augment the app with some additional theory-based components that we thought were missing, like habit theory. And we did extensive development work with patients and clinicians to get their feedback, and we finally finalised the intervention materials. So then that brought us to the next stage in the MRC framework, which is piloting and then evaluating the intervention. And that's where we're at now. We were lucky enough to get 4 years of funding from Yorkshire Cancer Research to conduct a pilot of our intervention versus usual care in 60 breast, prostate and colorectal cancer patients. And if it seems feasible, we have inbuilt funding to go on and do a full randomised controlled trial. And this time, based on patient feedback, we're actually recruiting patients during and after treatment, and we have quite extensive screening for safety before we enrol them. Our primary outcome is an objective measure of physical activity, accelerometry, and we have a number of patient-reported outcomes as well. However, as with other studies, we're now sort of subject to the coronavirus and delays. So we hoped to start recruitment this year, but we've been delayed, and we're hoping that we might be able to start next year. So it's kind of watch this space. But I would really like to thank a number of my collaborators, and I'd like to welcome any questions.
Thank you very much for a great presentation. Are there any questions from the audience? There's one raised hand. I can't see.
So it's Geeta Ramdari. Can you hear me?
Yep.
Yep. Hi. Thank you, Abby. That was fantastic. It was really nice to see those development phases in this an area of real interest. One question though, you commented where, sort of in that early work about the ethnic diversity of the people who responded to the surveys, and we know that this is a major issue with recruitment to research, and NIHR have been doing some work on this. And did you get a feel where, because we know that, for example, if the target population is London, there's 40% of people are not from white backgrounds, and it is a very diverse city. Did you get a sense whether or not the 13,000 you sent out, whether or not there was diversity in that population? Is this an issue of people from certain backgrounds not accessing these specialist cancer services because of health inequalities, issues with access, etc.? Because of course it's going to have an influence further down the line, isn't it, about who these sorts of interventions work with and who might be missing out?
Yeah, it's a great question, and I wish I had the answer, but our recruitment method meant that we couldn't access participant details unless they returned surveys and consented. So we actually, because the hospital sites were responsible for doing that, and that kind of allowed us the mass mail out, but what it didn't allow us to do was have data on people that didn't respond. So I don't have the answer, but it's a great point, and it's a huge problem, and it's something that I think we really have to address. I mean, there are obviously potential language barriers, but that's definitely not the only thing that's going on. So I don't know, and I'd welcome any thoughts, really.
Yeah, there is something we're wrangling with as well, but it'd be good to maybe have a think about that. Thank you, Abby.
Are there any other questions from the audience? No, I'll just say that I can see how this would be useful in many different neuromuscular disorders, especially now with gyms closed and COVID-19. Any support, this looks like something that might be a good time also to be doing. I have a question about privacy of information. So my center is very obsessive whenever we collect data, where the data are stored and how do we ensure there's no leaks and that the participants' information are safe. So how do you base with an app? The data, do you have it in the cloud, or do you store it in your own servers? And are you allowed to use some of the data, not the one you collected for the study, but some of the other data that they might be using the app for future research down the line?
So in this particular study, we're actually not using any of the background tracking data because we really wanted to, and I think it would be really useful and important to do that. But it was really problematic trying to access that data from Public Health England. But patients they essentially look at the privacy notice of the app, and they have to consent to, if they're registering with the app, they have to consent to that privacy notice essentially. But I think if it's something that you want to embark on, work really closely with the app developers and try and sort of identify a way that you can get that background data, because obviously, so we're having to measure adherence using questionnaires, which feels completely wrong when you've got a digital methodology. So I don't know if that answers your question. But I think essentially in other app studies that we've done, we've got our legal team to review the privacy notice, and if participants are consenting to it, then that's fine. And any identifiable information that we collect as the research team, we obviously have very strict protocols for storing that.
Okay, thank you. Are there any other questions? I see Rick has an MSG t-shirt there.
Sorry, yeah. I figured I couldn't have an MSG meeting without an MSG t-shirt. So I may put it on tomorrow on the weekend.
Looks
Sorry,
good.
that wasn't a question.
So thank you very much for a great talk. We're doing really, really well on time. So I'm going to do another small housekeeping issue since people are being a little shy with questions. We are having some breakout sessions, some breakout rooms later after the second half of the morning. Some of our sponsors are having some networking sessions, so I do encourage everyone to try to log in and have a chat with some of the sponsors. You can hear about the upcoming studies and things that are coming out. for the flash presenter. So normally we'd have a poster room and we will all be having drinks and talking with the presenters and commenting on the posters. We will be having flash presentations in the next following day. So some of the poster presenters will be giving a short talk on their poster, but I do encourage you to try to go to the breakout sessions and then they can interact with each other. And the whole idea is to try to get more senior researchers and clinicians interacting with the more junior ones to give more, to give feedback. So I do encourage everyone to go to this breakout sessions. So we have, we're running 5 minutes early. Gil, what do you think? Do we take a break or shall we just continue, Rick?
Well, Rick is not here yet, so let's
yeah.
wait 5 minutes.
No, I'm here. I'm here. I came back. Just getting coffee.
Not Richard, Rick.
Oh, our speaker.
Yeah.
Well, that's.
Well,
Oh,
So let's just take a 5-minute break, and we'll be right back with Rich.
got it.
okay.
Hey, Gil, how many people are signed in watching?
We have 94 attendees, and they can hear us right now. So.
Yeah, I know.
Oh, okay. Just so you know.
Well, you have 94 plus 117.
Well, there's 23 people on the backstage, and then 94 people in the audience. So 117.
But it's weird that we had almost 500 sign up. So I hope I guess they'll tune in off and on over the 3 days.
I suspect that's the case. It might be hard for some people to stay for the whole chunk of the morning. So I suspect people will be logging into some of the talks that they are mostly interested. Well, there's something coming in the chat.
I think this is working pretty well.
I think, yeah, I mean, I still miss the human interaction, but I think it's working fairly well.
Yeah, no, I agree. It's good, Rick.
Dr. Howard, if you click on the participant at the bottom of your screen, you can see.
I've just enabled it for the audience. It was set so that they could not see the participant list, but.
I'm sorry. Okay, good.
Oh, and we have Rick here. He's connecting.
Yeah, we'll give him a couple of minutes just in case people went to have a toilet or coffee break.
Okay, sure.
Liz, there's a question on the chat regarding late registration, so I'll leave you to answer that.
Is that possible?
I'll reply.
Yeah, Goran is he was one of my residents, and he's a big shot neuromuscular disease. So let's see if we can let him sign in.
Hi, Rick. Thank you for joining us.
Hi.
We'll give him a couple of minutes just to go back on time. We were a little early, so we gave people a tiny break.
Okay.
So just to make sure people come back just in case they step out of the computers.
Hi, Dr. Chapman. I'm the other Rick.
Oh, hi. Nice to meet you. So to share my slides, I just will use the share screen option.
Yeah, you could pull it up now if you like.
Okay, let me.
So I hate to do a do you know thing on this, Dr. Chapman, but do you know the cardiologist David Cohen, who does economic analysis of cardiovascular studies?
Yes, I do. We did some work together
Yeah,
back.
I figured it's a small world.
A graduate student, yeah.
He was here in Columbia the last 2 days. I'm trying to entice him to move here.
Oh, okay. He would be a good catch.
Yeah, he's good.
He's good, yeah.
I don't think there's a whole lot of you folks in the world that do this very detailed economic analysis. I'm really looking forward to your talk.
It's a small club, yeah.
Yes, and tomorrow, for people who are interested, we have someone coming from Canada, from CADES, to talk also a little bit about health economic assessment.
Okay.
So it's just part of the planning committee. We thought, all these new medications are coming, and very exciting times for also very expensive medications. This is a subject we all will have to be more and more involved. So this is part of the reasons we wanted you to come.
Yeah, it's important, I think, to figure out how to pay for these before they get to the market so that people get access as soon as they're available.
Okay, well, one minute. And
okay.
You can pull up your slides now, Rick.
Are you seeing them now?
It's loading. There we go. Looks good.
Good.
Perfect. So I'd like to welcome our next speaker, Rick Chapman from ICER. For those of you not familiar, that's the Institute for Clinical and Economic Review. It's one of the agencies that conducts health technology and health economic assessments, which I'm sure we will all be more and more familiar as we get more treatment. And he will talk about the health economic assessment of Spinraza and Zolgensma for spinal muscular atrophy. Rick?
Thank you. And good morning to everyone. Today I'm going to talk about ICER, or the Institute for Clinical and Economic Review's health economic assessment of 2 treatments for spinal muscular atrophy, Spinraza and Zolgensma. So first, I just want to acknowledge that this model was originally developed with some partners at the University of Sheffield, Matt Stevenson and Praveen Thakala, and Varun Kumar, who was formerly at the Institute for Clinical and Economic Review. And I just want to mention I have no conflicts of interest relevant to this work. So first, a little bit about the Institute. We're an independent health technology assessment group. We develop publicly available value assessment reports on medical tests, treatments, and delivery system innovations. And at the end of our assessments, we convene regional independent appraisal committees to have public hearings on each report. And for example, yesterday, we just had a public hearing on treatments for ulcerative colitis. And all of these reports and our meetings are archived on our website so that people can use those as time goes on. So a couple of years ago, we in March of 2019 is when our report came out, we wanted to look at treatments for spinal muscular atrophy. And we follow a similar process for all of our reports that starts out with scoping, with guidance from patients, clinical experts, manufacturers, and other stakeholders. We try to cast a wide net and talk to as many stakeholders as we can to find out what's going to be important to look at in an area, both clinically and from a patient standpoint. Then we conduct a systematic evidence analysis looking at the literature and randomized controlled trial findings. And along with that evidence analysis, which we conduct, as I mentioned, we worked with the University of Sheffield to do cost effectiveness modeling. We think it's important to look at the cost effectiveness of the treatments as they come to market. We publish our findings in a draft report, which we put out for public comment, and we often make revisions to that draft report, I would say almost always, based on the public comments we receive. And as part of that, we also have expert reviewers take a look at a report, both before and during the drafts phase. And and then finally, we come out with an evidence report following that draft and revision process that is intended to support our public meetings where we have voting around what we found and policy discussions around what may be important to ensure that patients can get access to treatments in a timely manner. This particular report was published in March of 2019, as I mentioned, and we've subsequently put together a manuscript on some of the findings from that report, which is currently in press. So our objective for the economic evaluation part of this assessment was to look at the lifetime cost effectiveness of Spinraza and Zolgensma, each compared to best supportive care for treatment of spinal muscular atrophy. And we use a lifetime horizon to make sure that we can get all the downstream cost and impacts in our analysis. The 2 treatments we looked at, as I mentioned, were Spinraza and Zolgensma. Spinraza at the time had been approved. It was approved in December 2016. And Zolgensma at the time was investigational. It was approved in May of 2019. But as I said, when we did our assessment, it was still under assessment by the FDA. Part of the reason, and we often hear that we should wait and do our results later when more data is available, especially when more real world data is available, not just clinical trial data. But we like to time our assessments around the time of approval because we know that's when important decisions are being made about coverage and prices for drugs. And if without this information, we think those decisions would be made in more of a vacuum than with this information available. So the scope of our review was any population that was eligible for these treatments. At the time, Zolgensma only had trial in type 1 SMA patients, and so those the more severe patients, and so those are the ones that I'll be focusing on here. And our key outcomes were not only survival, but also other milestones such as the need for ventilation or motor functioning and other milestones that may be important to patients. And of course, we wanted to also look at any potential harms from these treatments. So in brief, we created a new model which had two separate phases. One was short-term and really replicated what we saw in the trials and matched the duration of those trials. But then we also did a long-term model which extrapolated outcomes beyond that. And you'll see that that was, one of the more difficult parts of this analysis was figuring out how to do that extrapolation. We took a US healthcare sector perspective. As I mentioned before, we thought it was important to look at a lifetime of outcomes and cost. We did discount those at 3% per year to account for net present value. And we looked at both cost per quality adjusted life year, taking into account not only length of life, but also the quality of life in different health states. But we also did a cost per life year gained, where we did just look at the life years that were gained by the treatment. So our model was based on some broad motor function milestones that had been reported in the trials. We also thought it was important to account for mortality, of course, in this kind of a condition. Permanent ventilation, we knew, was an important outcome for patients to avoid. And one of the wrinkles to this assessment that made it more difficult was that Zolgensma was a single-arm study. So we had to use best supported care data from the Spinraza studies as a stand-in for what would have happened had there been a control arm there. And I just want to mention this. This is something we're seeing more and more, especially with gene therapies for ultra-rare diseases, is a lack of comparator, which often leads to a need to use some type of historical control when we evaluate these types of treatments. This is just a schematic of the model that we used for type 1 SMA patients. I won't go through this in great detail, but you can see we had the short-term study data using published time points within the trial and the outcomes at the end of the trial. And then we had to do long-term extrapolation in terms of where patients would move from these different health states: permanent ventilation, not sitting, sitting, walking, and of course there was a chance of death from any of these health states. And then long term, what we were really trying to track was, did patients avoid permanent ventilation, and what was the impact on mortality? So you can see the pretty broad health states here. We knew there were certain impacts of these treatments on patients that these broad categories might not get. So, somebody might be in the not sitting health state, but still have some good outcomes from the treatment in terms of being able to move better and have more control over their mobility. And so we did try to account for that later, and I'll talk about that as I go through this. These were the studies that we had available at the time for our evaluation. And you can see that the studies that are highlighted in red were non-comparative. And so you could see for Zolgensma, all of the data we had was non-comparative. And so that led to some difficulties in knowing how to model this, and also made it difficult to do any direct comparisons between Zolgensma and Spinraza. So there were some additional issues other than just the non-comparative data that we also had to deal with. There was potential confounding of some of the Zolgensma data because some of these patients were receiving Spinraza at the same time after they had received Zolgensma. 5 of the 12 patients started Spinraza treatment, and 2 of those discontinued, but we didn't know the reasons for that. We also had to, as I mentioned before, extrapolate long-term survival from some of the other shorter-term data that we had from trials. And so we had to make some assumptions about different health states and what the survival would be there. In this particular case, we set the not sitting health state as equal to survival on permanent ventilation in the absence of other data. And I mentioned before, we were concerned that these broad health states we were using were perhaps too broad and weren't capturing some benefits that patients might be getting from the treatment that would happen within those health states. And so we just made an assumption that patients were getting that benefit and bumped up the quality of life for patients in those health states as though all of them were getting that benefit. Another major hurdle in our analysis was that, as I said, Zolgensma was not approved at the time, so we didn't know what the price would be. We did have some signals from Wall Street analysts and from the company itself in terms of what they were thinking about would be a fair price for this drug, and it was in the range of several millions of dollars. We assumed a placeholder price kind of in the middle of that range of $2 million. So in our results, we found for Spinraza, the incremental cost effectiveness ratio in our base case of over $1 million per quality and about $590,000 per life year for Spinraza. However, for Zolgensma, even at the assumed price of $2 million, we found much better cost effectiveness ratios because of the potentially bigger impact on patients. We found ratios of $240,000 per quality and $182,000 per life year for Zolgensma. And for those that are not familiar, often the range that's quoted for the US is $50,000 to $150,000 per quality or per life year is considered as cost effective. So you can see that at $2 million, Zolgensma was approaching what people would consider cost effective in the US. And decision makers are often willing to, for rare orphan diseases, and especially ones that have such severe impact as SMA, are often willing to accept higher cost effectiveness ratios. So I think most payers would consider Zolgensma to be cost effective at that price. And the price that we found that would make it cost effective from $100,000 to $150,000 per quality was $310,000 to $900,000. So a pretty big range, but you could see $900,000, is a fairly substantial price, but would still be considered cost effective under our base case assumptions. And was not too far off, I think, from the final price, which was around 1.2 million. And as our key policy recommendation coming out of our assessment and our public meeting, we knew that we're going to be dealing with treatments like this that have new mechanisms of action. They're often very new types of treatments like gene therapies, and so we're not going to have a lot of long-term safety and efficacy data for these types of treatments. We also know they're often going to be one-time treatments and have very large impacts on patients in terms of both quality of life, length of life, and potential savings from healthcare costs that are avoided. And so we knew that these treatments could potentially be very expensive and still look cost effective, like we saw here with Zolgensma. And so we thought in those types of cases, it's going to be reasonable for insurers and other payers to think about the outcomes-based contracts or other types of novel payment arrangements that would put some of these cost of treatments at risk until it's been assured that patients are receiving the sustained benefit that we think they're going to get from these treatments. And we thought that was important to point out, because our concern is that without thoughts about these types of novel payment arrangements, there could be issues that some of these important, very revolutionary treatments wouldn't get to patients in a timely manner because of squabbling over access and financial issues. We knew that because of a lot of the uncertainties and issues I talked about before, that our base case was subject to a lot of assumptions. So we thought it was important to do sensitivity and scenario analyses, varying those assumptions and using different inputs. We found that for Spinraza, the results were not very sensitive to changes in those inputs. Still did not look very cost effective. However, for Zolgensma, again, this is using the assumed $2 million price. We found that there were several scenarios, and the majority of our probabilistic sensitivity analyses where the cost effectiveness ratios were below $250,000 per quality or per life year. And so that we found that it would be probable that Zolgensma could be cost effective or approaching cost effectiveness for this price. There were limitations, as I mentioned before, in our analysis that we had to try to overcome. Lack of long-term data. There's a lot of uncertainty about future outcomes. And I think especially for things like gene therapies, which are relatively novel, I think there's even more uncertainty about what future outcomes could be, whether they'll be sustained, whether there may be safety problems in the future. And one of the things that we've run across with these types of therapies that we don't deal with most other treatments is there's a concern that some of these gene therapies are irreversible and may preclude future gene therapies that would target the same areas. And so there's a concern that early adopters may be penalized because they're not able to get later gene therapy treatments that may have more sustained or broader benefits. We knew, as I said earlier, that there are broad health states could miss some of the benefits that are associated for treatment. So we tried to account for that by being conservative and erring on the side of the treatment and giving it the benefit there, even though we didn't know that all patients were getting it. And as I mentioned, we did extensive sensitivity and scenario analyses to explore the impact of some of these assumptions. As I mentioned, our assessments have this public comment phase where we get comments from different stakeholders. And in this particular case, we did receive comments that people thought there would not be an increase in survival when entering permanent ventilation for the people receiving these treatments. And so to account for that, we did change some of the inputs we were using there to make sure that our model was consistent. There was a belief that the health states in our model were too granular to detect important benefits to the patients. This again is the broad health benefit. And that's when, in response to that, that's when we added in this extra utility benefit for these health states. There were still some controversies and uncertainties when we presented these results at our meeting. Some people thought we were being too generous for Zolgensma, given the narrow eligibility criteria for their trials and the very small sample size. There were very few patients at the time who had been treated. And that was a real balancing act when we're thinking about how much weight to give to what seemed to be enormous benefit for the patients who had been treated, but the uncertainty given that there was no comparison and it was a small sample size with relatively short-term data. Long-term safety and efficacy, as I mentioned before, there were a lot of questions around that. It was difficult to compare Spinraza and Zolgensma directly because of the lack of comparative data. And the possibility of combination or sequential therapy also casts some doubt on our findings. It was unclear whether people with Zolgensma would go on to have Spinraza treatment as well if they didn't see the outcomes they wanted as quickly as they wanted. And it was unclear whether people would use Spinraza and then go on to Zolgensma or how that would play out over time. And finally, people pointed out to us that for a disease like this, which affects children, the caregiver burden was very important. We did consider including caregiver quality of life in our assessment, but that is a fairly controversial and not well laid out methodology in cost effectiveness analysis. And we were concerned that in some cases, this could even lead to counterintuitive results in that keeping a patient alive in a state with lower quality of life and that required caregiving might be seen as a burden when we didn't think that was fair to caregivers or patients. We also knew that there were other benefits and contextual considerations that would not be captured either in our clinical review because they weren't captured in clinical trials or in our cost effectiveness analysis because they couldn't be captured in that way. And so some of those benefits were that this was the first FDA approved treatment that would modify disease progression, Spinraza was. Zolgensma, as I mentioned, is a promising novel gene therapy, so that people were very excited about that. It was a one-time intravenous injection, so it really reduced the burden on patients in terms of treatment. There were, these improvements in patients' motor functioning could allow greater ability for self-care and independence, things that are very important to patients and their families, like return to school or work, but that would not necessarily be captured in our analyses. And there was possibility that there were impacts beyond the healthcare sector that would not be captured in this analysis. So in summary, we were able to do economic assessments for these treatments. It was difficult and did require a lot of assumptions. But we think that this was a good proof of concept. It was one of the first gene therapies we had looked at. We know more of these are going to be coming along, and they're going to raise specific challenges. In a lot of these rare diseases, there's a lack of standard patient-centered outcome measures. We often have to use surrogate outcomes, and it's difficult to know how those are going to translate into patient-important long-term outcomes. There's often no standardization of what usual care is for some of these diseases, and there's often not a control arm in the trials that we're looking at. We don't know long-term impacts of some of these treatments, and the small population sizes for a lot of these conditions make it difficult to conduct randomized controlled trials with control arms and with large numbers of patients. And so we know that these are difficulties we're going to have when we look at any of these types of treatments. All of those things led to more uncertainty in our assessment of the clinical effectiveness, and in turn that translates into more uncertainty in our cost effectiveness analysis. Quality of life for these diseases, and especially ones that impact young children, is often very difficult to measure, but we know it's often a very important impact of a treatment. And I mentioned the caregiver and family impacts are often very important, but hard to know how to handle in these assessments. And the patterns of health spending for patients are often not known. The natural history and what will happen over time, especially as these patients live longer, is often speculation. So as I mentioned, our meetings are recorded to our website. We published our final report after a public meeting on this in March of 2019. And it includes descriptions of our deliberation around the findings here and a policy roundtable discussion. And I'll stop there and open it up for questions.
Thank you very much. There is a question coming in the chat. I'm going to read it out loud. From Russell Butterfield. Can you discuss how models might be different with pre-symptomatic treatment? In the NURTURE trial, 23 out of 25 patients met independent ambulation, so potential outcomes, including lifespan and comorbidities, are very different from ENDEAR. similar, if not better outcomes are expected from pre-symptomatic treatment with Zolgensma?
Yeah, we did actually include a scenario analysis where we tried to look at pre-symptomatic SMA. At the time, we didn't have any data for Zolgensma in that population. So we really kind of made some assumptions. And I think in the report, we actually called it drug X because we wanted to make clear we weren't actually saying that this was what Zolgensma was going to do in that population. But we sort of made an assumption that if we had similar impact as we're seeing in type 1 in these patients, what would happen. And it was, actually very promising. We found that, again, Zolgensma would look very cost effective there. I don't have the results right here in front of me, so I can't remember what we found for Spinraza, but I believe it also looked more cost effective, although not as cost effective as Zolgensma in that population.
And there's a follow-up question. It says, can you address limitations of QALY as a tool for analysis for treatment of a lethal disorder given at birth that results in what is likely a normal lifespan? Is there a better way to model a definitive treatment?
Yeah, that's a very good question. It's one we struggle with a lot. The quality adjusted life year is used in a lot of health systems for these types of health economic assessments. It's kind of the gold standard in the field. At the same time, we know that there are flaws in the quality. It doesn't capture everything that's important to patients. And there are people who disagree with the quality completely and think it's discriminatory because it does look at quality of life. And so people with lower quality of lives, they think are being valued less than people with higher quality of life. We like to point out that, well, yes, it does take account of quality of life, but the main way it's being used is to look at improvements in quality of life. And so the more deficit there is there, the better the chance that a treatment can show improvements in quality of life along with length of life. But we know that it's not capturing everything. That's why we include cost per life year as well, because we know, especially for diseases like this, which have big mortality impacts, that that gain in length of life may be what's most important to patients, much more, or to stakeholders, much more so than the quality of life improvements. We also include in all of our assessments, not just the cost effectiveness, but also a review of the comparative clinical effectiveness on its own. And we also include in every report discussions from patients and patient groups in terms of what's important to them and what their unmet needs are, as well as those, that slide I showed on potential other benefits and contextual considerations. That's a chapter in every one of our reports that really tries to flesh out a lot of these things that we know are maybe very important, but can only be captured in a qualitative way and are very difficult to quantify. And in our public meetings, our panels, in considering the evidence on these, take into account all of those qualitative aspects as well as the things we're able to quantify.
Okay, we have lots of questions coming, so to move fast so we can answer as much as we can. From Sarah Jones, how soon after the new Witherspoon data will be published do you expect ICER to perform a review?
We, in terms of our initial reviews, we try to publish those, as I mentioned, around the time that we think a drug is going to be approved, because we want that to perhaps influence pricing negotiations and coverage policies. So that people have all the information they need to do that. We know that if we waited later, we might have more data, but at that point, a lot of those decisions have already been made and are kind of set in stone. We are now trying to do updates on our reports. So annually, we're going to be looking to see if there's additional data or evidence that would make it important to do an update. And then we're going to try to put those in the queue. Unfortunately, we don't have the resources to be able to update every review that we've done, which we would like to do if we could.
Okay, and I think we have a couple more minutes. Valerie, I'm gonna ask you to unmute because we had several points, but from Valeria says, interesting approach. Just wanted to point out that their overview was rather ungenerous towards a couple of points. Small cohort, there are more than 10,000 people on Spinraza worldwide now. The pre-symptomatic kids in the Nurture study have been followed for almost 4 years now. And this is important, has not been done for the initial studies on Zolgensma. And finally, probably best to compare the pre-symptomatic in one cohort than the other, and that would be fair. Anyone want to address?
Yeah, I think those are fair points. You know, we were working with the data we had available at the time. And as I mentioned, that was one of the real things we were trying to balance is how do you balance the larger numbers of patients who had been treated and studied with one treatment over a longer time period with, you know, a small uncontrolled number of patients on the other treatment that had a much shorter follow-up, and there was much more uncertainty around, but also seemed to be having a much more dramatic impact on patients.
So we're right on time. Thank you very much, Rick, for this great talk. And us, we're going to be moving to our next speaker, but I would call all our community, all these areas of uncertainty, this is the research we should be doing in patient preferences and arm control group. This is the data you're going to need for further review. So it opens a whole set of questions for us to answer as researchers.
Thank you, and thanks for the opportunity.
Okay. Jeff, hi. Are you good to go? Are you
would it
good to
be
go?
possible?
Like
Hi,
we have,
are you hearing me okay?
yes.
Okay. Would it be possible for me to use my own slides, as I actually did a couple of tweaks today?
Sure.
Not a problem. Feel free to share.
Okay, thank you.
Okay, let me introduce you properly then. So keeping back to incorporating and implementing new technologies, Dr. Gus Curran from University of Arkansas will talk about us incorporating implementation science questions into clinical effectiveness trials. So you have the floor.
Okay, and so I can share my own screen now?
Yes, you can. Feel free.
Okay, I'm hitting the share screen and oh, here we go. We are good. I'm going to share that. And I'm just going to go to the full view if that's going to work okay. Okay, so is that going to work for us?
It's loading. Give us a second. All right, we're good.
Okay, great. So thank you so much for having me today. Here is the title slide. And so what we're going to be talking about is, you know, ways to think about and to integrate implementation science type questions into clinical effectiveness trials. Okay. And so the goals for the session are as follows. So like I just said, you know, trying to, you know, talk about ways to working in these kinds of questions into clinical effectiveness trials, but also to contemplate these kinds of issues even possibly prior to starting a clinical trial. We're going to talk about the concept of hybrid designs, which combine elements of clinical effectiveness and implementation research questions into the same kinds of studies. A few years back now, we proposed a range of 3 such hybrid designs. We are going to focus on what we call the type 1 approach, which is exploring implementability, if we could make up a word here, of an intervention while we are testing its effectiveness. And I'm going to close with just a quick slide on this concept, which is sometimes called in my field designing for dissemination, which deals more with preparatory to starting a new trial, working on intervention components to try to make them, you know, as best set up for uptake as possible. So first, I figured that it might make sense for us to spend a minute or two, you know, on this notion of, okay, well, what is implementation science research anyway, as some folks here, you know, might, you know, be less familiar. Here's a very common and, you know, formal definition, which is getting at that we are talking about that it's the scientific study of methods to promote the systematic uptake of research findings and other evidence-based practices into routine practice. Okay, so the scientific study basically of how best to support uptake. When I teach about this stuff, especially with folks who might be new to the field, I often try to use some fairly non-sciencey language as follows. So I will often refer to the intervention evidence-based practice that's being supported as the thing. And clinical effectiveness research, which you guys are familiar with more, I think, you know, looks more at whether the thing works, whether it's having the desired effects on patient symptoms and functioning. Implementation research, however, looks more at how best to help people and places, meaning providers, clinics, systems, how best to help them to do the thing, okay? And in our field, we have this term implementation strategies, which really refer to these interventions, or as I say here in the super sort of lay language, the stuff that we do to try to help the people and places to do the thing. And there's a lot of research now on varying types of strategies, which seem to be helpful with promoting uptake of a wide range of practices. It's things like education and training, clinical reminders, use of audit and feedback, training and using local champions, coaching, problem solving, facilitation, all of those kinds of things. And just to close the loop here, the main outcomes which we often focus on in this kind of work in layperson's terms refer really to how much and how well that these strategies are working to help these people and places to do the thing, to implement the intervention to the extent that we want it to be delivered to qualified patients, but also that it's delivered, to the quality that we would like it to happen. So when do we do implementation research? And I'm sharing here kind of a traditional pipeline, which was certainly, common about 20 years ago when I first got started in this field. And there was very much this notion that we need to finish or do a lot of efficacy work, showing promise of interventions in highly controlled trials, move into what we often call effectiveness research, where we're moving into more real world contexts. We're having interventions delivered by the routine clinicians and staff in those places. We want to show strong effectiveness there. And then, and possibly even only then, will we start to then talk about implementation research questions and try to do some studies now on how best might we need to support uptake of this evidence-based practice in real world settings, okay? I would argue now that in the past 8 to 10 years, a lot more people in our field are talking about ways that we can blend effectiveness research and implementation research, combine questions from these two areas with the goal of trying to speed translation, trying to not be so siloed, not be so separate, but to plan ahead for some of these issues while people are still focused to some extent on clinical effectiveness. And so that's my focus today. So what sort of questions are we talking about combining in these kinds of combo hybrid studies? So these are questions that I think that, you know, most of the folks in this group are usually focused on. Clinical trial questions, you know, having some intervention which you're trying to test whether it beats some control or comparative intervention, trying to understand for whom does it work better and under what conditions, and also to some extent maybe for some of your work trying to look at cost effectiveness, and, you know, trying to get a sense of possibly comparing interventions, you know, which might be more cost effective. These are some of the kinds of questions which I'm talking about, which can be integrated into clinical effectiveness research to help us to plan ahead for future research, which might be more focused on helping uptake, you know, after we have a fairly good sense or as we are learning about effectiveness. So questions like, how implementable is the intervention itself, and what ways might it need to be adapted for better uptake in the future? What barriers, facilitators might be happening in these contexts where you're testing these interventions? And we often try to get folks to think about these kinds of barriers, facilitators, which are happening at multiple levels in a clinical system where these are being tested. So what system level barriers, clinic level barriers, stuff about the inner and outer setting, workflow, you know, staffing roles, policies, mandates, provider level issues around knowledge, attitudes, level of support and prompts needed, but also at the patient level. You know, how ready, willing, and able are patients to engage with this practice, and then next to start to think about strategies which might be needed to support optimal uptake of these interventions. Okay, so these next couple of slides, I'm going to be talking about you know, how we might go about combining these kinds of questions in the same studies. And this was our first paper from 2012, which first laid out these 3 hybrid design types. We have a newer paper which kind of brought some of this thinking forward with some, you know, new recommendations, and these are out there, you know, for your reference. So in the first paper, we floated 3 types of these hybrid designs: hybrid type 1, hybrid type 2, and hybrid type 3, which varied in their extent of the emphasis that is being placed on the effectiveness side or the uptake side. And I'm going to be talking mostly today for the rest of the talk about the hybrid type 1 design or hybrid type 1 variant, which is still largely a traditional clinical effectiveness trial, but we add in some specific and systematic work to try to learn about implementation issues which are happening during the trial, and we contemplate with our stakeholders during the trial how things might need to be revised, to be adapted for better uptake in the future. These other hybrid types, deal more with either in a type 2, more of a dual focus on testing some intervention against a comparison while also testing some strategy package to help with uptake compared to some other one, or hybrid type 3, which is more of an uptake trial where the main outcomes are more about the strategies to help people to do the intervention, but we are still collecting data on these clinical outcomes. So here's a sense of the makeup and structure that is really common in these hybrid type 1 designs. So again, it's largely focused on testing a clinical intervention normally in a patient level randomized trial, but there's this other part which is focusing on the factors related to uptake. And I have this indication here of 80% versus 20% with the 80% of time and effort largely focused on the clinical effectiveness side, but the smaller amount here focused on these uptake issues. These things operationally normally work like a conventional effectiveness study, but we add on what we often call an implementation-focused process evaluation. So in that part of it, you're trying to describe the implementation experience that is happening during the trial, what worked, what didn't, barriers to strong uptake during the trial, to ask questions and to try to learn about how the intervention or how it's delivered might need to be adapted going forward to increase uptake potential, and also trying to explore potential strategies which might be needed in the future to help people in places to do the thing in the real world after we have a real good sense that this intervention is strong. How do we help people to do it in the real world? Okay, so remember, all effectiveness trials use quote unquote implementation strategies to help with the delivery of this intervention. We just normally don't call them that. And in most of these trials, these strategies are normally pretty resource heavy. We pay clinics to take part. We often pay interventionists. We often pay for the care. We do frequent fidelity checks. We intervene when that is not working so well. And we have a sense that some or many of these strategies used to help deliver the intervention during these trials might not be feasible for supporting widespread adoption in the real world when you don't have grant money to help with those kind of things. But our central point here is that we can learn a lot about the use of those strategies during these trials to help us to plan for the next step where more real world strategies might be needed. I will talk here about a quick example from a study that we called CALM some years back. So in large part, it was a large clinical effectiveness trial of an anxiety intervention in primary care. Big study, four cities, 17 clinics, 1,000 patients. We trained care managers who were normally nurses or social workers who were already working in those clinics to learn to use a software tool to work with patients to navigate through this treatment manual. And starting towards the end of the trial and going in to the post phase, we did this relatively strenuous qualitative process evaluation to try to, ask a number of stakeholders, varying questions about how things went during the trial, what they would need to possibly keep doing the intervention after the study is over, and what naive clinics who weren't part of the study might actually need to adopt this. And so we did about 50 interviews with providers, nurses, staff, front office, the interventionists from each of these 17 clinics. And most of these interviews were done on the phone. They were relatively feasible. Our qualitative interview guide was guided by a framework which helps us to know what kinds of uptake factors to ask about. And, that's a very necessary component for these kinds of studies. What did we learn? We learned a lot of stuff, but I'll kind of cut to these points here. So I think that these data illustrate, I think the crucial value of this kind of work. So what we learned through these interviews was that most of the providers in the participating clinics did not end up referring a lot of patients for the trial, and some referred nobody. Now, how this worked is, or how the goal was, is fairly common in these kinds of studies, where the study team goes to these clinics, they talk with all of the providers about the study, they try to get them motivated to take part, to do the screenings, to get a sense of who might be good candidates for this intervention, and to refer patients for the trial and to be randomized. So we did a focused qualitative analysis of providers who referred a lot of patients and those who didn't. And we found that those who referred a whole lot of patients had a pre-existing interest in mental health issues, and already kind of saw the value of this kind of intervention. Those who didn't refer a lot of patients were not persuaded during the site trainings that this was a good enough idea to actually take part. So using some of the formal language from my field, we were seeing here that uptake and reach of this intervention during the trial was not great, even though the research team tried to get all providers to refer. So a key barrier to future uptake of this intervention is buy-in at the provider level. The standard strategies which we had used during the study to try to get them motivated and excited didn't seem to work. So moving forward, we have to work harder at that aspect of it if in the real world we want to see high levels of reach where the highest number of qualified patients are actually being referred to and get this information. And so if we had done this kind of work separately, and so this stuff wasn't part of the effectiveness trial, and we started to ask these later as part of the earlier questions on uptake focused studies, we would have learned this, late. So there's a lot of examples of these kinds of studies out there. Here's a few that I like. I'm happy to share these and more, and you will have access to my slides. My last slide here is just sharing this notion of another way to approach this called design for dissemination, which is floating this notion that even prior to developing some intervention, you might want to spend some time thinking about these implementability questions and trying to build components that based upon some end user feedback about ways that it could be developed initially to maximize its uptake potential. And there are frameworks which can be used in this hybrid type 1 post-hoc analysis to learn about those factors. You actually can do some of this work prior to building your intervention or prior to adapting some evidence-based practice for a new population or a new context to get a head start on trying to operationalize these components to maximize their uptake ability. So I'm going to stop there, and I'm happy to take any questions which you might have.
Wonderful. Thank you very much. It's really, really, really useful for us. There's a question from Nadeem. Any thoughts given on how to improve enrollment for minority patients?
Yeah, so I think that it makes sense as early as possible to try to bring in perspectives from multiple stakeholders. You have the more provider system side and strategies that they need to help support the system to offer it. But I think you also need to involve stakeholders from the patient side to help them learn what education, training, marketing strategies that they might need to help them be more ready, willing, and able to engage with this intervention. And if you are dealing with interventions which carry a lot of stigma, if you're dealing with marginalized populations with a lot of barriers to access to care, it's all the more important to have as much feedback from those folks as possible during the whole process.
Wonderful. We have another question from Geeta. Is this mainly the domain of complex interventions, for example, behavior change, or is this important for drug trials too?
I think that the more complicated the intervention is, I think this kind of focus might be more important. But I think that there are times when we underemphasize the complex factors that go into medication trials, and especially more in the real world. Trying to prompt providers to, you know, realize who are the appropriate candidates or not, to navigate drug choices, to be aware of, you know, dose ranges, complications with other medications, trying to deal with issues like, you know, cost and coverage and patient preference and shared decision making. I mean, all of these come into play in those kinds of studies too. And, you know, I think that there is a role for this kind of stuff in those too. And if you don't focus on these, that it could be at your peril, or it could certainly slow the translational process later when these become more of the focus, if we move into more rollout research supporting widespread uptake.
Wonderful. Thank you very much. And we're out of time for more questions. Thank you again for a wonderful talk. And that brings us to our last speaker for this morning, well, at least here for the first session. And this is Dr. Sabrina Paganoni, who will talk about the Haley, I'm hoping I'm pronouncing it well, ALS platform trial, innovative trial design and collaboration to accelerate drug development.
Okay, great. So I'll ask you to advance the slides. So apologies again for the glitch. So essentially, I'm going to cover these topics today. Next slide. And as I was saying earlier, you know, it could be interesting to see whether this framework might be of interest also for other neuromuscular diseases. There is a lot of interest in platform trials in the neurosciences right now. The concept originated in cancer, and as you may know, it's actually being organized for Duchenne muscular dystrophy, but certainly could apply to other disease areas as well. Next slide. So why was this a good idea for ALS at this time? Because ALS is the neuromuscular disease with the largest drug pipeline. This is based on IQVIA data that was collected in collaboration with the Muscular Dystrophy Association. And we then updated it with now over 160 companies in the ALS space. Now, compounds might be at different stages of development. Obviously, some are more early stage and others are ready to go into the clinic. But essentially, the point here is that a platform trial approach makes sense when there are several investigational products in the pipeline. And so it makes sense to try to accelerate the drug testing field so that we can actually make progress faster, because obviously our patients don't have time to wait. Next slide. So we actually quantified sort of the time efficiencies and also the impact on number of participants required to find effective treatments. So let's say this is a simulation from our biostatisticians. So let's say that each investigational product that we are testing has a 10% chance of being positive, of affecting disease progression. So then it would take us 10 treatments to find the first effective treatments. Now, if you can click one more time, you'll see that in an ALS traditional trial, it would take us over 10 years to be able to test all of these treatments sequentially with a relatively large number of participants involved, 50% of whom would be on placebo. Now, if you were to use this to test the same exact 10 treatments, but use a platform trial approach instead of a traditional approach, you would dramatically cut down the time it takes to test all these treatments and the number of participants required. Importantly, you can significantly cut down the number of people on placebo. So as you can see on the slide, in our simulations, this was really phenomenal savings all across the board in terms of time, effort, number of participants requested, and most importantly for our patients, reduction of the need for placebo. Next slide. So in a nutshell, how does this work? So when you test drugs in the traditional clinical trial paradigm, you test one drug at a time. And that's what we've been doing for many years for good reasons. It's a system that works, that can provide, answer one question at a time for each treatment, and so then you move on. However, though, when you have multiple drugs that are available for testing, that's not an efficient way of testing the drugs by any means, because it's almost like always being in construction mode. You have to basically build an infrastructure to test the one drug, answer the one question, and then dismantle the infrastructure and move on. So we took a dramatically different approach that we learned from cancer. So in the platform approach, you want to build your infrastructure at the very beginning of the trial, build an optimized clinical trial protocol that can accommodate multiple treatments, and then begin testing them using and reusing the same infrastructure over and over again, so that instead of just testing one drug at a time, you can test multiple at a time. And most importantly, you can keep the infrastructure open long-term, so that you can continue to test more drugs as they become available. And so that's exactly the infrastructure that we built for the Healy ALS platform trial. So we spent quite a bit of time learning from people in other fields of medicine who had set up successful platform trials. And then we worked together as a community with many ALS investigators providing input, so that we could really build what we think is an optimal protocol for testing drugs at this time. And so we launched the trial with 3 treatments, and now we're gonna add more, and I'm gonna explain how that works. Next slide. So this is also a way to be able to do something that our patients have been asking us to do for a long time. Again, less placebo, more access, and more options, because the truth is that it's only a minority of ALS patients that really participate in clinical trials. So we wanted to change that. So how does this work? Well, in a platform trial, what happens is that when the participant enrolls in the platform trial, he or she will be assigned to any of the treatments that are being tested at that time in the platform. Let's say that there's 2 or that a third is added. So the participant is assigned randomly to any of these treatments, but they will know to which treatment they might, to which treatment they are assigned. So they will know, for example, that they are assigned to treatment A. Now, what happens then is that within that regimen or treatment arm, the participant will be randomized to either active or placebo. And that's the placebo-controlled period of the trial, which in our case is 24 weeks or about 6 months. But within that regimen, as you can see in the graph, the randomization ratio is in favor of active. In other words, for every 4 people who are assigned to treatment A, 3 will go on active and only 1 on placebo. So obviously it's great gainings. So I think the animations don't fully work well. If you can, yeah, thank you for clicking. So basically what you see here is that at the end of the regimen, which is placebo controlled, then all participants can either enter an open label extension or re-enter the platform trial if they're still eligible and be assigned to another drug. So essentially with this shared infrastructure, you're able again to minimize the chances of a participant being on placebo, and you give them more access and more options, especially because they have different options at the end of the placebo-controlled trial, and they could potentially be re-enrolled and reassigned to a different treatment. From the point of view of a drug manufacturer, you still maintain statistical power with a 1-to-1 randomization because the data from the placebo participants are shared. So drug A, the green one, for example, will be compared 1-to-1 active to shared placebo. So you still maintain statistical power. That's obviously important. Next slide. So it took us a few months to, again, bring together the community, multiple stakeholders listed on the slide. And we recently launched the trial and launched enrollment in July. And enrollment is going incredibly well. So I'll tell you more about that in a second. Next slide. So the design of the trial was made possible thanks to the commitment of an engaged trial design committee. I'm sorry, all the names are not showing for some reason on the slide, but and if you can go back. So we had essentially a core team of investigators listed at the top, several consultants from both MGH and Berry Consultants, and a news advisory panel that I'm sorry, not all the names are showing for some reason. I'm not sure about the technical problems today, but essentially we had a great advisory panel with multiple investigators from NILS. Some of them are not listed, but really we leveraged the expertise of several in the ALS community that contributed their expertise in terms of design design and scientific aspects of the trial. Next slide. We also leveraged the NILS clinical operations team that includes experts in project management, data management, and capture grants monitoring outcomes from both MGH and the Barrow Neurological Institute. Next slide. And we leveraged the NILS sites, as you know, a clinical trial network with over 20 years of experience and several studies completed to date. So we selected 54 of those sites to be part of the trial, governed by the NILS Central IRB for maximal efficiency, and hopefully we'll be able to expand the number of sites very soon. Next slide. Yeah. Something that's very important to us is the engagement with the patient community. It's been absolutely fantastic to work very closely since 2019 with a committed group of patients that advise us on every aspect of the trial and actually continue to provide amazing input in terms of, initially the design, but now the execution and ways to connect with patients and the community. Next slide. So in order to select the treatments to test first in the trial, we issued an initial request for proposals that was met with great interest from industry and investigators. And so we received several applications, almost 30 from 10 countries. 5 drugs were selected to enter the platform now, and the selection was made by a committee that includes scientists from the Healy Center as well as the NILS Scientific Advisory Board. I want to emphasize that we continue to accept nominations for new treatments, because after the first 5, we certainly want to continue to add more treatments and test more. As a matter of fact, we are already in engaged discussions with 3 additional companies for 2021. And the link there is to submit nominations for new treatments. Next slide. Of note, we have had a great relationship with the FDA. The 2 pictures are from our 2 initial meetings in 2019. Our 2020 meetings were virtual, so I don't have nice pictures from those, but we continue to remain in communication with the FDA. We received a May Proceed letter for the master protocol at the beginning of the year, and then we continue to work with them on new regimens. Next slide. So to summarize, we built a trial that we think offers several benefits over traditional trials, and really this is supported by all the stakeholders that I mentioned so far, including regulators, industry, patients, and investigators. And this is sort of a snapshot of the main features of the trial. And now I want to explain a little bit more about the drugs that we are testing and the target population. Next slide. So the target population for everyone is the same, for every treatment is the same, and it's listed here. I'm not going to go through the details, but essentially we have a core set of key inclusion and exclusion criteria that define our target population. And then in terms of each regimen or each drug, they need to follow the same criteria so that the target population is the same. That's critical to allow placebo sharing. The only additional criteria that might differ from regimen to regimen, and I want to emphasize the differences are really minimized, are only for selected safety issues. So for example, if there's a specific drug-drug interaction with a specific investigational product, that drug-drug interaction needs to be obviously part of the protocol, and so those participants that are on that drug might not participate in that particular regimen. But by and large, the target population is the same. Treatment duration is the same for every regimen, and the size of each regimen is the same. Specifically, the treatment is for 24 weeks plus the open label extension for each regimen, and the sample size per regimen is a total sample size of 160 participants, most of them on active and some on placebo, as I explained earlier, and then the placebo data is shared. Next slide. The endpoints are also the same for all regimens. As the primary endpoint, we use the ALS-FRSR adjusted for mortality, and we discussed with the FDA that if there was a very significant effect on the primary outcome, this trial could be used as a pivotal trial for registration of a new investigational product. We also collect additional secondary endpoints, and I want to spend a second about the exploratory endpoints, which I think are also a very important feature of the program. Next slide. Now, because we are using a shared infrastructure and we continue to accumulate placebo data from the different regimens, we essentially also have a continuously growing natural history study. And so we really wanted to take advantage of that to contribute to ALS science by do deep genotyping and phenotyping and collecting additional biomarkers from all participants. So for each regimen, that allows us to have additional exploratory analysis of interesting biomarkers for that particular drug, but also importantly, because we're using the shared infrastructure, we can share the data with the ALS community in terms of the placebo data so that we can really learn more about the behavior of these biomarkers in the growing placebo cohort. Next slide. So this is where we started with 3 regimens. And again, I'm sorry for the slide, as for some reason there's some formatting issues here, but we're starting with 3 treatments: Zellucoplan, Verdiperstat, and CNM-Au8. And again, that's the schematic that I explained earlier. Each of them will also offer an open label extension. These drugs do have very different mechanisms of action, and it's actually good to include, you know, for our you know, trial that targets the entire ALS population. It's not only a genetic subtype or a specific subtype. It's good for us to test drugs with different mechanisms of action so that we can learn more about you know, each pathway and each you know, each different mechanism of action that might be of relevance to the disease. We are adding the 4th regimen, predopidine, soon, and we're in discussion with 3 additional companies for 2021. Next slide. So then COVID happened in March, as you all know. We were about to start enrollment in March, April, and the week before we were to hold the investigator meeting in Boston. Actually, we had a few cases in Boston, so we had to turn the investigator meeting to a virtual setting. We were able to do the investigator meeting in March. Next slide. And then we were able to continue thanks to all the sites. I know many of you are on the line. All the sites really were dedicated and committed to the trial, and they worked through the pandemic, working remotely, doing everything possible to ensure the safe start of the trial. And so we started enrollment in July. We have 21 sites open so far. The other ones are getting very close. So the goal is to activate all 54 sites very soon. And there's huge interest from the patient community. So the enrollment numbers keep changing, but we're now, I believe, over 90 participants enrolled in just a couple of months. So, and basically 50% of the sites open. So really fantastic progress. And we will continue to update the patient community on our enrollment. We're going to start a series of webinars very soon where we continue to update everyone on enrollment progress, because really I think we're going to actually exceed enrollment expectations. So it's really a fantastic time to really accelerate the development of new drugs for ALS. Next slide. Coming soon also, we are working on building an expanded access program in parallel to the platform trial for patients who are not eligible for the trial. This is not available yet, but there is great interest from the community, and we're working on sort of how to best set this up. We're working also, again, to achieve this goal from many different perspectives, and so we'll hopefully more to come on this very soon. Next slide. So to conclude, I hope I share that platform trials can greatly accelerate the clinical trial development of new drugs for ALS and other diseases. There is strong support for this approach from multiple stakeholders, and the Healy ALS platform trial was built to be a perpetual trial so that we can continue to add drugs. The link to our website is there, you know, we continue to accept nominations for new compounds, and hopefully we'll be able to expand to new trial sites as well. I do have one extra slides that I asked if I could share, and I was told it's okay to put a plug here because we're expanding, again, expanding so fast. We are looking to recruit new ALS clinical trialists to work at the Healy Center in close collaboration with us. I have here the names of the people who are leading the search, Mary Sukovich and James Berry. That's the email. Also feel free to contact me. My email is at the bottom with any questions about the process or about the platform trial. So thank you for your attention.
Wonderful. Thank you very much. We'll move right to questions.
Yeah.
The first question is there any issue with patient selection bias? sorry, my screen closed. With patient selection bias, when it comes to the open label phase, as patients can choose to move to a different trial.
Yeah, so the primary endpoints and really sort of to test the effectiveness of the drug, really that's based on the placebo-controlled portion of the trial. Now the open label phase can be useful in terms of collecting some additional long-term safety data, but obviously that's not the primary trial to establish effectiveness. Again, there's many different things that can affect the open label phase. So I would say again, we discussed with the FDA that the trial, if positive, could lead to registration of a new investigational product, and that comment relates to the placebo-controlled portion of the trial. Now, the open label phase, again, is to accumulate long-term data in more advanced patients as well. From a patient perspective, yes, they can choose to either contribute to that scientific goal of long-term safety in a broader and more advanced population, or they can choose to, if they're still eligible, presumably they're still early in their disease, there is a cutoff of 3 years from onset to enter the master protocol, and then they can actually go and be essentially a new patient, a new participant. I hope that answers your question. Let me know if not.
We can comment further if we have time. So there's a couple more questions. One one from Michael McDermott. I'll read it, but you can also see it in the Q&A. Can you comment more on the implications of using a shared placebo group? If you compare drug A versus the shared placebo, some who are in the shared placebo group are being randomized to that group potentially much later in calendar time and may not be comparable to people who were randomized to drug A. Is that not a problem? It might be less of a problem if all drugs were being studied simultaneously in calendar time and randomization was to all drugs and placebo, but that does not seem to be the case here.
Yeah, no, that's an excellent question. So I guess you're getting to the problem of concurrent controls versus historical controls, and that's absolutely part of what we're paying close attention to here. So first of all, we are, so there's, I'm not a statistician, so I know you are a statistician, so obviously it would be good for you maybe to connect with our statisticians who have been involved with several other platform trials and build all the models. But essentially, there is, the model accounts for concurrent versus non-concurrent controls. So that's taken into account, and the models for drift over time. And so for now, the idea is to use all controls, but if we see that there is a drift, we might not be able to use sort of the older controls for the new regimens. The goal here is to also always have multiple drugs open at that time so that we have a large enough pool of concurrent controls anyway at all times. Plus, again, we have the accumulating controls that the model monitors very carefully to make sure that there's no drift there. And also the weight depends on the timing. Sort of the more recent controls contribute more to the comparison. I hope that answers that question. Our statisticians can certainly explain that much better, and we can have follow-up conversation.
Okay, and there's another question. Can patients request to switch or reassign to another regimen prior completing 24-week course?
No, they can't. They cannot be assigned to another regimen within the 24-week period. So they can obviously choose to stop study treatment, and if they do so, we will encourage them to continue follow up under the ITT principle in that particular regimen, and then they have to wait until their projected 24-week last visit to be able to re-enter the protocol and be rescreened for re-entry into the master protocol. And again, that's because we cannot, to avoid bias, we cannot allow patients to basically switch midway through the trial. If participants can also choose to stop drug and not come back for study visits off drug, and that's fine, but they still have to wait until their projected 24-week course would have, completed.
Wonderful. And just Michael McDeckerman's answer to your answer, he was perfect. Many thanks.
Thank you.
Okay, so I think that's it. We're right on time, which might be one of the good things about a virtual meeting, I guess. So we have a break. We have a 15-minute break for the second session. Remember that the session starts at 11:10 Central Time. So we'll see you back later. Thanks, everyone.
Hi, Shafiq.
Hi, Reza. Very excited for your talk.
Thank you.
Hi, Reza. Rick.
Hi.
Hi, Shafiq.
Hi, Rick. This conference has been great so far. I'm really enjoying it.
It's working. It's
It's
amazing.
working.
It's working. Don't have as many people signed in as we have registered, but maybe we'll see what happens over the next 3 days.
I think they come and go. I do
Yeah.
have some other people here. I have Dr. Colin Quinn also with me listening to the talks because I think we hit the limit and we couldn't get him.
Well, there was a 500 person limit. That's what we had our Zoom. That's what we paid for the Zoom fee. And if we would have gone over there, it would have been a lot more expensive. Now, next year, we can anticipate and budget that in.
Yeah, that's amazing. Okay, so I'm gonna introduce Dr. Reza Sajjari. He's gonna be talking to us about proximal nerve imaging in CMT1A. Reza is an MSG fellow. He's also an assistant professor of neurology at Harvard and Mass General Hospital. So Reza, there you go.
Hey, thank you. Let me see if I can share my screen. So you have my screen?
We do, yes.
Perfect, thank you. So it's been a great honor to be the fellow of the MSG and present in this year's meeting. I'll present updates on the project of the proximal nerve imaging in hereditary neuropathies, specifically CMT1A. So in brief, CMT1A is the most common known type of hereditary neuropathy, despite recent improvements in our understanding of its pathophysiology and novel therapeutic venues. There is paucity of reliable outcomes to detect changes over time and in response to potential treatments. So far, imaging and neurophysiology markers that we've been working with represent mostly distal peripheral nerve function, and these have failed to produce sensitive outcomes to detect changes over a short period of time. In comparison, and hypothetically, proximal nerves, structures such as sciatic nerve or nerve roots maintain better structural and functional integrity in a presumed length-dependent process and probably are more sensitive and responsive. We now have more psychometrically devised clinical and patient-reported outcomes with improved sensitivity and responsiveness, most notably CMT-FUM and CMT-PEDS. And muscle MRI, more specifically, calf muscle fat fraction has been proposed as a potential imaging biomarker similar to other neurophysiologic measures. However, calf muscle fat fraction likely also represents a degree of axonal loss in more distal distribution. Our colleagues at UCL were successful showing reasonable sensitivity and responsiveness of calf muscle fat fraction in patients with CMT1A in a year study. So clinical imaging has some value in diagnosis of tumors, inflammation, compressive processes. And enlarged nerve roots in cervical or lumbosacral imaging are often overlooked by general radiologists unless very prominent, like this case. And they reported as acquired or hereditary process without any specificity or sensitivity. Beyond clinical imaging, which is mostly limited to T1 and T2 sequences, research MRIs are primarily reliant on different diffusion and volumetric techniques. So I'll give a very summary of what has been going on in the world of imaging, especially in the recent past year or so, and then we'll give you an update about our study. So volumetric measures and signal intensities have been utilized to look at peripheral nerves in CIDP and CMT. There's been equivocal data to support a possible association between disease severity and nerve volume in CIDP, but there isn't a stronger association between disease duration and nerve volume in patients with CIDP, interestingly, regardless of the status of disease or its severity. In a recent study of 32 patients with CMT1A, and I think there were 13 healthy controls, nerve volume was shown to be significantly different in patients with CMT1A for the whole sciatic and tibial nerve. The sciatic nerve volume modestly correlated with the whole set of clinical scores, and interestingly, no association with the tibial nerve volume and the clinical scores. Animal models have been used to show the histopathological association of DTI measures, such as fractional anisotropy, which I'll mention in a second, with axonal integrity in the sciatic nerve. These techniques showed fairly accurate biological correlation to transient ischemia and axonal density. And MTR is another metric which is a macromolecular content of, measures macromolecular content of the nerve, more representing the myelin lipid composition and more sensitive to changes in the myelin density. A more detailed axon myelin measurement using higher gradient imaging allows better estimation of axon diameter and density, and in summary, axonal integrity. What's most important for us in porcine optic nerve and sciatic nerve on the left panel, mouse corpus callosum in the center panel, and recently in human spinal cord on the right side. Diffusion measures were obtained from a group of patients with CIDP, showing strong correlation between distal nerve compound motor action potential and fractional anisotropy of the tibial nerve. And previously, significantly lower FA of sciatic nerve and DTI measurement was shown in 15 patients with CMT1A compared to healthy controls. In a more recent study, 18 patients with demyelinating CMTs, most of them I believe had CMT1A, and controls in very low gradient MRI of the lower extremities participated. They were able to show the DTI signals could discriminate between the demyelinating nerve pathology of CMT patients from normal nerves, somewhat better than volume. Interestingly, they also showed slightly better discriminative validity in more proximal portions of the sciatic nerve. Similar to diffusion MRI, MTR, briefly mentioned, is sensitive to changes in myelin density, which can occur in demyelination or axonal loss. And Dr. Lee's group previously looked at a selected group of patients. I think there were 8 of them were had CMT1A. They showed significant difference between signal intensity in patients with CMT1A from healthy controls. On the right panel, you see the calculated MTR representation in patient CMT1A compared to the control. The goal of our study was to develop and validate non-invasive imaging biomarker more sensitive to change and detect changes in sciatic nerve of patients with CMT1A. We hypothesized that accurate estimation of axonal integrity and numbers in more proximal nerve structures with more preserved function could serve as discriminative and evaluative outcomes, strongly correlate with neurophysiologic, metric, and even other imaging biomarkers, and better detect changes over time and predict response to changes over time, disease progression. So primary aims of the study was to show the difference between healthy subjects and patients with CMT1A and assess correlation between axonal integrity and other clinical and imaging biomarkers, including calf muscle fat fraction. And the third aim was to evaluate longitudinal changes in these biomarkers over time in relation to other outcomes. For the first phase of the study, we planned to scan about 20 patients in healthy controls, which were age-matched with sciatic nerve imaging, calf muscle imaging, and CMT-specific clinical outcomes. In the following cold development in initial pilot imaging and quality control, our recruitment was put on hold from February, March of this year. So these were preliminary measures from our very first control subject using the high gradient 3T scanner and functional anisotropy maps. After a long delay, we eventually started rescanning patients with healthy controls and using both optimized 3T scanner that we had developed, the Coil 4, and 7T MRI scanner. We have so far scanned about 3 patients and 3 controls, or 2 of them are sort of with the new protocol. And we've used a 4-channel 3 Tesla MRI and a 28-channel called 7 Tesla MRI. We were able to show better imaging quality of 7T versus 3T imaging in both anatomical and diffusion imaging sequences. These images are from the same healthy control. You can see the 3T sequences in the first row compared to the 7T sequences from the similar level. In addition to higher resolution, you note the improvement in the diffusion sequences, which obviously allows for more accurate measurement of axon integrity, number, size, and even density. You can see the axial view of the sciatic nerve in a healthy control on the right panel, and in a patient with CMT1A on the left panel using 7T scanner, you can see the larger nerve volume in the patient with CMT1A. And here you can see axial views of the sciatic nerve in a patient with CMT1A on a 7T MRI, and reconstruction of the sciatic nerve with tibial and peroneal bundles. It's not very difficult to appreciate and measure almost individual bundles and the tibial and peroneal divisions of the sciatic nerve. Here you can see the axial sciatic nerve of a patient with CMT1A, about approximately 10 centimeters above the knee. The bottom panel shows axial sciatic nerve of a healthy control from the same level. You can appreciate the different signal changes in T2 relaxation, in ADC, apparent diffusion coefficient, and FA sequences in the patient and the control subject. T2 relaxation tells us how fast the MRI signal dies out due to individual atoms interfering with one another, often seen in the setting of disruption of the cell wall or integrity or even inflammation. A higher T2 relaxation has been shown to be correlated with inflammation in prior studies and disruption the structure of muscle and nerve and even muscle. ADC or apparent diffusion coefficient tells us how freely the atoms randomly move around. Similar to T2, it tends to be higher in more disrupted structures when atoms can move more freely alongside the nerve structures. And we can estimate ADC with much better resolution than prior studies. And the FA is essentially an extension of ADC, but the idea is that the movement of atoms is largest along the axis of the nerves or the fascicles, but more constrained perpendicular to its axis because the atoms will bump into the boundaries of the nerve or fascicle. A higher value means that the atoms only moving along one axis is suggesting of better axonal integrity. So you can easily appreciate the difference between FA and ADC measurements in patients with the CMT1A and the healthy control. So we are still very far from making solid conclusions based on very small number of patients that we have so far been able to obtain, and this very prelim data, but it would be interesting to make some small observations. The top panel shows axial sciatic nerve of a 40-year-old patient with CMT-ES score of 12, and the bottom panel shows axial sciatic nerve of another 40-year-old patient with CMT score of 8. The first patient has a slightly weaker finger abduction and plantar flexion, and slightly higher sensory level to both pinprick and vibration compared to the first patient. You can appreciate the slight difference in the T2's relaxation and FA in these two patients. So a longitudinal study will be helpful to look at changes over time and responsiveness. We are maximizing our recruitment efforts to include at least one subject per week. That's what we've been doing for the past month. And we plan to apply for support to proceed to the next phase of the study, which will be longitudinal phase, and look at the possible collaborations. So there are multitude of technical issues and challenges requiring support from radiologists, physicists, and other colleagues. A major concern has been related to meaningfulness of or content validity in psychometrics language of these measurements. We hypothesize that there will be significant change in imaging biomarkers over time, but we know that hereditary neuropathies are very slowly progressive processes, and currently used outcome measures lack responsiveness. So it's plausible to appreciate changes in imaging biomarkers despite none to minimal changes in the other outcomes or disease severity measured by other outcomes. So while this may suggest better responsiveness of imaging outcomes without definite clinical correlations, at times it may be difficult to argue for better association. So again, I'm very grateful for the opportunity, for the great mentors and colleagues and support from Muscle Study Group, American Brain Foundation, and Inherited Neuropathy Consortium. Thank you.
Fantastic talk and subject, Reza. Uh, we had, I think Jiaoshin had one question. Is that correct? But I couldn't see it.
Let me see.
He raised his hand. But
I've asked him to unmute.
okay. So Jiaoshin, can you unmute and you can, and then
Ask
you
your
have,
question then.
and ask the question, please.
We also have Zicarito. I'm sorry, Ricardo. You can also unmute. There you go.
Hi, do you hear me?
Oh, yes. Hi.
Hi, Raja. Nice to see you. Congratulations for your presentation. Really interesting. I have just a question for you. If you have some data from other clinical outcome assessment and the MRI. Thank you.
So, yes, so we are, for these patients, because of the current limitations with our ability to bring them to sort of more elaborate evaluation. So we have we have obtained the CMTNS score because these patients are also part of the INC 6601 study. So we have their clinical data and information. We have measured their ankle dorsiflexion and using a dynamometry. We were planning to do some neurophysiological measurements too, but that's currently on hold. But we are getting also calf muscle fat fraction in MRI and some other basic clinical metrics. Yeah.
Thank you.
Welcome.
So we have another, somebody else who wrote a question, Chris Haven. It would seem other muscle diseases could benefit from the study, or I guess neuropathy disorders, maybe he meant. Are you looking at expanding, since there are several imaging studies, for example, in FSHD?
So this is an interesting. This was an interesting opportunity for me because I was interested in imaging the nerve, but the collaborator I have here is interested in imaging the muscle. So I said, well, it's an interesting combination because currently we have this relatively better defined outcome measure looking at the muscle fat fraction in patients with CMT. It would be a good thing to combine so we can use your resources to scan patients while I have these patients from my side, and it would be a good correlation combination study, and we can use a better technology to show better sensitivity. I know that there are studies that my colleagues are doing with FSH and looking at the muscle, but we would be interested in, I'm sure my collaborator would be definitely interested in collaboration, but definitely, yeah.
I think Vera, Dr. Brill, please unmute yourself and you can answer, ask a question.
You're on, Vera. No, we can't hear her. We'll have to come back to her later. We're actually out of time. Oh, there we go. There we go.
Okay. So my question is, thank you for an excellent presentation. And I'm just wondering, do you see this as a way to look at early efficacy with an intervention, if you had one, that would predict with prolonged therapy that you might have clinical improvement? Because if you just had a change in imaging and there was no functional correlation with it, it might be difficult to convince others of the efficacy of an intervention.
Exactly, that would be part of the conceptual limitation of the studies like this. So if you're defining a new outcome, its meaningfulness is questionable. So whether this is something that is going to be, you hypothesize that maybe this is more sensitive and showing the axonal loss at an earlier stage compared to the other outcomes. But conceptually, you cannot make promises, at least now.
Okay, thank you, Reza. So we're gonna move on because we're running out of time here. We're gonna transition to now the flash presentation. So these are short, because these are submitted abstracts that we're going to be discussing. The first one is going to be with Dr. Francesco Muttoni, long-term safety and efficacy of golodirsen in male patients with Duchenne muscular dystrophy amenable to exon 53 skipping. So looking forward to your talk, Francesco.
Hi.
Hi,
Hi.
Francesco.
Good afternoon. Let me see if do you see the right thing?
We don't see anything yet, so.
Okay. Yeah, maybe this then.
It's loading. Start the presentation.
Yeah, you
there
have
we
it?
go. You're all set.
Okay, thank you. Well, thank you for asking me to make this presentation on behalf of the SKIP-NMD consortium. This is to present for the first time aspects related to the long-term safety and efficacy of golodirsen in children with Duchenne receiving this drug to make exon 53 skipping. In terms of disclosure, this is the disclosure of myself and of the other SKIP-NMD consortium members. The sponsor of this study was Sarepta. So this is a phase 1/2 ongoing multicenter study that in the first part had a dose escalation, dose titration, where a group of children received either golodirsen at 4, 10, 20, and 30 milligrams per kilo, and a group of other children received placebo. And after the data safety monitoring review, all children received golodirsen at a dose of 30 milligrams per kilo. And in addition, a group of another 13 children was recruited to increase the number of children receiving the 30 milligram per kilo dose. In terms of efficacy, we had two key efficacy measures. One was a biochemical efficacy that was comparing dystrophin between week 48 and baseline in all the children receiving treatment. And at week 144, we collected 6-minute walk test and forced vital capacity. The study also had an untreated arm, but this untreated arm was not genotypically matched and was to collect exploratory biomarker in Duchenne muscular dystrophy. And I will present now some data that have been recently published. These relate to the week 48 difference in exon skipping between baseline and the treatment and the children after treatment, with a 28-fold increase of the exon skipping of the right exon 53 skipped product. And of course, more importantly, the ability for this patient to produce some dystrophin with an increase from very low level at baseline to more than to a 1% with a 16-fold increase. And this was also followed by increase in dystrophin production measured by number of dystrophin positive fibers. And although not shown here, there was a correlation between the percentage of exon skipping and the dystrophin production in all patients. When it comes to the clinical efficacy, this was an open label study, and the need to compare data with genotypically matched control became clear as recent data show quite significant difference in the trajectory of patients with individual genotypes. And therefore, a post-hoc decision was made to compare the children receiving golodirsen to a matched external cohort of patients that have been recently published. This was a collaborative study between Italy, Belgium, and the UK, where 19 children with deletion amenable for exon 53 were compared. And you have here the age, the rise from the floor, and the 6-minute walk test baseline comparison of these children. So this is the 3-year 6-minute walk test data on the children in the control group in the natural history study. And you will appreciate this patient eventually lose 181 meters from baseline after 3 years. The patient receiving golodirsen also declined but the decline after 3 years was 99 meters with a difference of 82 meters compared to the external control. This is the loss of ambulation in patient, both receiving golodirsen in the umbra color or in the external group of patient. And maybe if you just concentrate on the 3-year column showing a difference between the external control with 26% heavy loss ambulation and 9% in the golodirsen treated patient. Regarding the respiratory function test, over 3 years, the patient on the golodirsen treated arm lost 8.38%. And the patient in the external control did not have forced vital capacity captured as part of the natural history study. And therefore, the comparison here is, if you like, less stringent, but is done with actually every single study on respiratory function decline in Duchenne muscular dystrophy in this age range that indicates an annual decline of percent predicted of 5%. So 8.38% over 3 years may indicate a difference. In terms of the safety and tolerability, there were no drug-related severe adverse event. There was no discontinuation related to treatment. On the whole, the treatment was well tolerated. And this is my last slide that suggests that we recently demonstrated and published the fact that golodirsen can induce skipping and protein expression. We now have perform an analysis, is a post-hoc analysis with the external group of patients with eligible deletion that are equivalent to the one that we are, the children recruited into the clinical studies. We showed here that trajectory of the ambulation and loss of ambulation that appear to be differing compared to the external control. I indicated that a limitation of the forced vital capacity, this was present in that particular group of patients, that this is not ideal, but there is no published data indicating that there is less than 5%, between 4.7 and 5% annual decline in every single studies. So that may also be of interest. Clearly, the drug was on the whole well tolerated with no toxicity. And we appreciate the limitation of the open label and external control of the study. But I think this is the first clinical efficacy data presented of this, and clearly ongoing randomized placebo-controlled study are confirming the validity of the finding that I reported today. Thank you.
Thank you, Francesco. Very, very promising data you showed here. Do we have any questions? We have a minute for questions. Okay, we do have one from Dr. Karen Stagelin. Does golodirsen cross the blood-brain barrier? Does it affect the cardiac muscle?
I think the golodirsen does not cross the blood-brain barrier. None of the morpholino, they are a neutrally charged molecule, cross the blood-brain barrier. And in terms of the ability of morpholino to target the heart, considering the work that has been done in preclinical model, in human, we don't know. Considering the work in preclinical model, one will remain skeptical. Is unlikely there is a significant targeting of the cardiac muscle with the current generation antisense oligonucleotides.
Okay, thank you very much, Dr. Francesco. We're gonna move on to satisfaction with ankle-foot orthosis in individuals with Charcot-Marie-Tooth disease with Dr. Ricardo Zuccarino. Ricardo?
Okay, do you hear me?
Yes, we can hear you.
Okay, I share my Okay. Okay, here we go.
It's good.
Okay, good. Thank you for the invitation. Good afternoon. And I just jump, okay, here. Ankle foot orthosis are commonly prescribed to individuals with Charcot-Marie-Tooth. And the aim of this study was to evaluate patient-reported satisfaction with orthotic devices and services in individuals with CMT to provide preliminary data for advancing AFO development and improving clinical care. We used the web-based Inherited Neuropathy Consortium contact registry for distributing this study survey. The contact registry includes nearly 3,500 individuals, and all participants between ages from 18 to 90 were invited and provided the consent. And then we just share out the OPUS, the Orthotic Prosthetics User Survey, and a open section in which we asked to the patients what would they change on their AFOs and eventually limitation of their AFOs. OPUS, we decided to choose the OPUS because it's a reliable and valid patients reported outcome measure, and designed to evaluate patient satisfaction with both devices and the clinical services. Includes 11 devices specific questions and 10 services related questions. We had a lot of answers, almost more than 300 individuals just completed both the subscales of the OPUS, and here you can see the demographic characteristics of our respondents. Over one third of individuals just provided negative responses about their AFOs, and specifically they just underline some problems with the appearance of their AFO, but also they experience in discomfort and especially with pain, with abrasion and irritation of the skin. And another point just came out was the cost of the AFOs aligned with the other previous studies that are done with satisfaction with AFOs. About the clinical services, patients just indicated satisfaction with the services, but they just want to be more in the center of the decision pathway for just providing AFOs. We had some limitation in these studies because the total number of individuals that who use really AFOs is unknown, and it resulted for us in inability to calculate a meaningful response rate or just evaluate potential bias. Another point is the mean age, because is more indicative of older individuals of CMT. And so we don't know really about the younger ages of these patients. And also another point is that the use of the questionnaire also prevent other objective assessment of physical performance, device type, sensory impairment, and other indicators of disease, especially for severity or progression. But we think that this is a really interesting study because we had a lot of opinions of several hundred individuals with CMT and provide really interesting information for further study. I'm going to conclude my presentation, just underline that with this study, we had a lot of clear areas for improving our eventually prescription, and also for improving the EFO device comfort, especially for appearance, but also for reducing irritation and abrasion in the skins. And we think that, again, putting the patient at the center of our orthosis prescription pathway is the point for these patients, and just maybe continue research in these areas would be beneficial for AFO development, but above all for improving our clinical care. In the end, our study was published in July on muscle and nerve, and I have to thank especially to Professor Shai for his mentoring during my period in Iowa, at the University of Iowa, and a special thank to the Inherited Neuropathy Consortium group, and that's all. I'm here eventually if there are some questions. Thank you.
Thank you, Ricardo, for presenting this very important study. We have Gita Ramdari, who is raising her hand for a question. I'm gonna.
Thank you.
Go ahead.
Hi, Ricardo. How are you? That was really.
Hi,
really
Gita. How are you? It's nice to
was,
see you.
And it was really nice results. It was very interesting, and you know this is an area I'm interested in as well. And actually, it was interesting to see the results in that comfort and abrasions. You know, I'd expect we hear that clinically a lot, but I did wonder about the cost, because you noted that nearly 80% of the sample were from the USA and only 14 from Europe, and then smaller percentages from other places. Do you think this may have skewed that finding, because where in Europe then for instance in the UK people tend not to pay for their AFOs, they come through the health service. Do you think that might be why it featured more in your data set than perhaps if there'd been a bigger representation from other countries?
Gita, I don't hear you very well, but I understood your question. Thank you very much. I think you underline a very good point. I mean, the prescription pathway in the US is completely different than Europe.
Yeah.
And I think it's still, in any case, a problem about the cost, because eventually when we prescribe an AFO, even though we are in Europe, eventually there are some problems for patients just for reaching out eventually the correct shoes, for example, and this is in any case a cost for the patients, and I think it remain in any case a problem, and especially now in Europe. I don't know exactly in the UK, but in Italy it start the healthcare system just to cut off some prescribed furniture. And so sometimes especially when we prescribe specific AFOs, for example, in carbon or different type, more, I mean, high level, I mean, AFOs could be a problem.
Yeah.
But I agree with you that there is still more a problem in the US than in Europe.
Yeah, thank you.
You're welcome.
Ricardo, we have 2 questions, but we don't, actually 3 questions, but we don't have a lot of time.
I'm really
so
happy.
you can answer the question quickly. The first question from Dr. Gilwolf, analysis of difference between the different AFO models, materials, carbon versus plastic, etc. And then the question regarding, do you partner with PM&R physician in the US since they know much more than neurologists about orthotics? And then finally, a question from Maxwell Green, does this study affect your recommendation for type of AFO for patients?
Okay, I think I just summarized some of the answer in the limitation, because this is just a survey study. So a point and the limitation is that it's difficult just to understand exactly what type the patients have. And so also compare the different models in plastic, carbon, or what is the best in this type. And but this is our aim for future study, and for sure we want to correlate a different type of material of AFOs and eventually other clinical outcome measures that are coming up in CMT for CMT patients. So for sure we are going to underline this point. The second one was for the PMR in US. I agree that I mean PMR are more indicated for just follow this part of the prescription for CMT patients. I do think as well that the problem is also just increase the knowledge in the orthotic people. So maybe we could also in future collaborate for organize maybe a future just meeting focus on that. And the third, I don't remember. I don't remember the third one.
I think we're also unfortunately running out of time. So thank you very much for.
Okay. Does this study affect your recommendation for type of ACE to patients? I mean, we have no much data for answer to this question, but thank you very much for everyone and for your interest in my study.
Yeah, thank you, Ricardo. Next, we're going to have Al-Jawhara Alangari. She's going to talk to us about exploring muscle structure, function, and gait patterns in people with distal hereditary motor neuropathy, the natural history and the effect of rehabilitation interventions, as well as study protocol. There you go, Jawhara.
Hello, everyone. This is Al-Jawhara. Thank you very much for the introduction. And yes, this protocol is my PhD work, and it's aimed to explore the muscle structure, function, and gait pattern in people with distal hereditary motor neuropathy, or DHMN. Well, DHMN is one of the rarest inherited neuromuscular disorders, and it's characterized by a slowly progressive distal muscle weakness that manifests in the second decade of life. It's a very disabling condition, although it's not life-threatening, but affected people must eventually need aids to walk later in their life. So research in this area is needed to improve the quality of walking in people affected with DHMN. So we need to understand the natural history of the disease and to understand the impairments that lead to altered function, especially in gait, and to develop conservative interventions to correct walking gait. So the primary aim of this protocol is to explore the natural history of muscle structure function in DHMN. Secondary aims are to ascertain the relationship between intramuscular fat fraction, muscle volume, muscle strength, and moment power generation. Also to explore the effect of two commonly used rehabilitation methods, including carbon fiber ankle foot orthosis and resistance training exercises. We aim to recruit 20 people with DHMN aged over 18 and 20 matched control. We are planning to use 3 main outcome measures, including intramuscular fat fraction measured by MRI, isokinetic muscle strength measured by hemochrome testing and rehabilitation system, also kinetics and kinematics of gait measured by Codemotion system for 3D gait analysis. For rehabilitation, we will be using carbon fiber ankle foot orthosis bilaterally and resistance training exercises program for 16 weeks. So the study has the 3 main visits. At baseline, DHMN participants will undergo MRI scan, isokinetic muscle strength analysis, and 3D gait analysis. For direct comparison to identify deviation in gait, 20 controls are going to be recruited and will undergo the same measures. And to explore the effect of carbon fiber ankle foot orthosis, DHMN participants will undergo additional gait analysis in 3 different conditions. Once wearing their own shoes and wearing their own prescribed orthotics if they have one, and wearing carbon fiber ankle foot orthosis provided during the study. After 12 months from baseline, each participant will undergo the same measures again to explore the natural history of the disease. In the second visit, the DHMN participants will be invited to join the exercise trial. They will be eligible if they score muscle strength 4 or more on MRC scale on their ankle muscles. So after 12 months of resistance exercise program, same measures will be repeated to explore the effect of the training. So the findings collected from the study can help to evaluate the natural history of the disease and effectively provide guidance on managing DHMN. It also can help to improve patient outcomes in terms of walking gait and reduction of muscle weakness. And also, the data collected can help to calculate the effect size to conduct sample size calculation for larger trial in the future. That's it for now. Hopefully next time I come back with the collected data for this study. Thank you for listening, and if you have any questions, please feel free to ask.
Thank you, Al-Jawhar, for doing this very important study. Do we have any questions?
We're at the near the end of our day. I thought it's been a great morning. Are you still there, Dr. Hannah? I thought this has really been a great way to do the MSG. Dr. Mato, I'm glad you were there and weighed in, feisty as ever. Thank you. I was trying to chat with some of you and give you a hello, and I realized I was chatting to all of you. That wasn't my purpose, so apologize for that. I haven't figured out how to get some of the private chat things going.
There's Professor Hannah.
Are you there? Yeah, I was just saying. Yeah,
I'm still here. It's getting dark now.
Good. What time is it there?
7 o'clock.
Oh, okay. Dinner time.
Yeah, it's not too bad.
Yeah, all right. So the last thing of the day is there is now breakout rooms. Now, some of you have been doing Zoom meetings already. This is my first real Zoom meeting. I've been doing our neuromuscular course by Zoom with Katz and Levine and Dimachki, but it's a lot different. So I've never been in these Zoom meeting rooms, but Gil has sent you the link to get in it. And I've got the link too now. And I guess we're just supposed to
You
click.
could put up the information on how to do it.
What? I thought we just click on this link.
Yeah, well, but Gil's put some instructions together. He put up there.
Oh, it's more than just clicking on the link?
Yeah, well, there's instructions if you want to know how to do it.
All right, so when you enter the room, what you'll do is you will type in the chat. Basically, we'll have a room set up for each presenter. We have 3 people who will have rooms today. And you will enter in the chat what room you'd like to go to, and you'll be sent to that room. Now, when you want to change rooms, you will press the breakout room button, and then that'll pop open a menu that'll allow you to join another room. Now, if you're on your mobile phone, the instructions are a little different. What you will do is you will leave the breakout room that you are in, and then retype into the chat what room you'd like to go into next. But
So if we click on this link, which you sent us, what you're going to see then is this breakout rooms in progress page, and that's when you just click on what you want to go to.
So to enter the first room, I need to send you in there. You don't get that menu right away. So to enter the first room, you let me know in chat what room you'd like to go to first.
Oh.
I need a couple minutes to set that up, but we'll be sent to that room.
Oh. So you've got to set each person. Okay.
Or you could just start putting people in rooms and they could go back and forth, Gil.
Sure. Yeah, I could do that.
Well, but they may not want to be in that room.
I do have a separate room, sort of like a cigar lounge room, if people want to go in there first.
Cigars?
Oh, you're talking.
Is there a campfire there and whiskey?
Anything you want. Anything you want. Your imagination.
And then also Genentech has their separate Zoom links, and they're doing one-on-one sessions. And so I sent that information out, and we'll put it in the chat.
Okay, I'm putting a link to
Tony,
today's.
you can go to the Genentech room and
You have to go to their specific Zoom links, and Gil's going to put it in here for you.
So the link I just put is for the main mingling room. And now I'm going to put the 2 Genentech rooms.
all right. So when we click on those, we leave this meeting though, right?
That's right.
Yes.
All right. Well, I'm gonna hit the button and leave the meeting and see what happens.
Okay.
Michael, you have anything else to add? I thought this
No.
went well.
Yeah, it went really well, I think. So I'll see you in the chat room.
And thank all the committee, the meeting committee that moderated. Thank you, Carolina Shafiq, and thank you for all the presenters. That was really outstanding, really good. And then we'll convene again tomorrow morning at 8 Central Time.
Yes.
Okay. 2 p.m. UK time.
All right.
All right. See you guys there.
All right.
Bye.